Good morning and welcome to the ASAB Winter 2024 meeting. Thanks all so much for turning up, for coming along here. Many of you have come from all over the world, and that's really a great thing to see, particularly given the theme of the meeting for the next couple of days. So welcome to Edinburgh. You're seeing it on really quite some lovely weather. It's cold, crisp, and certainly when we were out last night, it looked beautiful. So I hope that you enjoy your time. If you're not from Edinburgh, that we can all enjoy our time in this city here. I'd like to thank presenters as well for all the preparations that you've made and for complying with the conference organisers, sending in your talks and things. That's been really helpful. Other people I'd like to welcome, particularly this morning, are the students of the Royal High School and Edinburgh College. So welcome. For those of you that haven't been to an ASAB winter meeting before, our first talk of the session is an open talk. It's one that's meant to be accessible to all. And so we've got some really exciting speaker. I'm going to let Claudia introduce a bit later on. But we invite local school children or students to come along for that. And then afterwards, you get a chance to sort of discuss what's going on and actually look around the posters as well. And so, you know, it's us trying to encourage, as animal behaviour researchers, we're trying to encourage, hopefully some of you lot, that in a few years' time, it'll be you giving the plenary talks up here. Hopefully we can inspire you to do that. So welcome for that. You'll have heard that I've been mentioning ASAB quite a lot. I hope that you're all well aware of it. I hope that you're all members. If not, can I encourage you, that's encourage with a strong E, to join up. Our meeting secretary, Anna Wilkinson. Where is Anna? Membership. Sorry, she's membership secretary. I'm meeting secretary. What do I know? She is welcoming people at the desk. If you want to join up, then please talk with her. We've got other members of the ASAB Council. That's the group of people that oversee the society here. We have got ourselves stickers. I know many of you have got stickers on your badges. We've got the sort of the round ones with two grebes with the ASAB logo on there. And so if you want to talk to any council members, please do approach us there. People of particular note that you might want to meet as members of societies to put faces to names. We've got Melissa Bateson, our president. So Melissa, if you could just stand up and wave. So Melissa leads us all. We've got Andy Radford. Where's Andy? Down here. Who is the editor of the journal Animal Behaviour. So if you'd like to talk to him about the journal, not probably about your specific manuscripts, but about the journal itself, if you've got ideas, then please do talk with Andy. And we've got Christos Ioannou. Is Christos? Hey, up here. So Christos, if you have had a travel grant or an attendance grant or a care grant to come to the meeting, Christos and the grants team have been responsible for that. So you may want to go and express thanks to him and just generally talk to him about opportunities for research grants, for attendance grants, care grants, and so on that ASAB provide. So for those of you that are not members of ASAB, ASAB is really the main organisation that brings together animal behaviour researchers in Europe and now beyond as well. We don't cover the Americas, that's our sister organisation there, but really many other countries. And ASAB hosts a number of conferences throughout the year, and it's through ASAB's generosity that we're able to make this meeting free to you all there. And we'll be talking a little bit more about that at the AGM, which brings me on to the point at 12:30 today. So after the second talk session, we've got the annual general meeting of ASAB. We aim to smash through it in 20 to 30 minutes. That's the ambition, which gives people an hour or so then to go for lunch. Clearly, if you're not an ASAB member, there's no obligation to attend. If you are an ASAB member, we do hope that you would join us for that time so that you can hear a little bit more about the society and ratify various decisions that need to be made, to hear the reports of the officers and to feel perhaps more of an involvement. There will also be, and this is a great moment, an opportunity for you to join ASAB in its leadership. So those of you who are members will have seen that there have been a number of elections going on for positions on council. We have one outstanding position left. That's mine. So I've been here for 6 years, and frankly, I'm only meant to be here for 2 3-year periods. We need someone to take over the meeting secretary, and we'll be talking about that at the AGM. If you're interested, please come and approach me and talk to me about that through the meeting. You won't miss me. I've got the foul pink shirt on. Okay, so I think that brings me to the end of sort of the ASAB side of things. The way that the ASAB meetings work, the winter meeting, is that although ASAB might pay for the venue and organise the catering and things like that, actually we have nothing to do with the science. The science is all led by teams of volunteers who put together a particular theme, work together and draw up a really, I hope you'll agree, a quite fantastic programme. And so this year we've been incredibly fortunate to have a great team of organisers led by Claudia, Vivek, Nicola, Chiara and Cedric, and they have a huge team of other folk behind them that have helped sifting through abstracts and so on. So on behalf of ASAB, I'd like to express our huge thanks to all those who've been involved in organising this meeting, and to hand over to Claudia, who's going to welcome you officially. But thanks very much indeed. Thank you.
Thank you very much, and thank you all for coming. It's a great pleasure. My name is Claudia Wascher, and I've emailed to all of you in the past. Can I briefly mention my team, and could you all please stand up? So Nicola, Chiara, Vivek, and Cedric, without whom, thank you, without whom this meeting would not have been possible. And we also have the little ASAB stickers, so please come and talk to us in the course of the meeting. A couple of introductory words. We have a very kind drink and reception sponsors of the Royal Society, and they have a desk outside, so please go to the Royal Society desk and check the materials they brought to us. And the Cambridge University Press quite kindly sponsors our poster prizes, so we will have ECR poster prizes, and the ECR posters are actually judged by our school students here, so you have a very important duty. And the prizes will be given on Friday during the closing remarks. This is our program. We actually had updates to our program very recently, so if you follow the program, please make sure that you have the updated version, which I sent to you via email, and it's also downloadable on our web page. A couple of words regarding socializing. So we will have a reception and poster session in the afternoon, but the venue closes at 8 o'clock in the morning. And in previous years, we had experience that people really like to socialize after this. So we actually suggest a Wetherspoon close to the venue, which can take large crowds. So we will be able to meet there. I would like to very warmly welcome also all our virtual attendees and hublets. So we have 4 hublets in France, Germany, Austria, and India. They've already logged on, so I know you're there. Very warm welcome. And over 200 virtual attendees, so it's great to have people all over the world joining our program. And in order for you to be able to interact with us, we have set up a Slido. So the Slido has the hashtag ASAB24. So if you like to ask a question, please log onto the Slido, type in your question, and we have one dedicated person in the room who will read out the questions. Regarding questions in the room, please make sure that you use the microphones. I appreciate that people sometimes have strong voices like I do, and we would be okay in the room, but for the virtual attendees to be able to hear you, you really need to use the microphone. So if you have a question, please stand up and use the microphones which you see on the corners there. For our talk sessions, we actually have something here. Sorry, the Austrian is in charge, so there will be very strict timekeeping. So we have a timekeeping charge. The chair will actually, the keeping person will actually show you when you have 2 minutes remaining for your talk, and will also show you when the time is up. And as a little incentive, we have a piece of chocolate, which you will only get if you keep to time. If you don't keep to time, the chair is allowed to eat your chocolate. Good. And last but not least, it's really our intention with this meeting to create a diverse, inclusive, accepting, welcoming, and safe space for everybody. So we do have a code of conduct which we everybody ask to follow. You can please read, please familiarize yourself with the code of conduct, and if there are any issues, please approach the organizing committee or any other person which is actually highlighted on the safe space posters outside. So if there's any reason to get in touch with us, please do so. There are posters around the venue to tell you how to do this. Having said this, I'll hand over to Chen now, who will talk a bit about social media before we start.
Hi, good morning. So, in case anybody noticed, we're moving over to Blue Sky. Last year was but a rumor, and now it is a reality. So please join and post on Blue Sky. Our hashtag is ASAB Winter 2024. I'll give you a quick rundown for those of you who aren't familiar with Blue Sky. It's just like Twitter. You can just sign up, make an account, blah, 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 but it's got a few other nice little features. For starters, it doesn't reward people being a-holes. So it's a much more positive space. So we hope that people will really find connecting with other animal behavior members much more rewarding. And all these little tips and tricks I'll have posted for you on our account there, so you can see all of this again. So you can actually block people and you'll never see them again. You can follow block lists, so you never have to see a troll again or a group of trolls that are harassing someone. If somebody decides to be a jerk to you about your post, you can simply not allow them to use your post to talk about you. But the best part about it is that we have topic feeds. So for this conference, we have our own feed. You can pin, and it will have everybody who uses our hashtag right on that feed, and you can very easily follow the conference. And then also, if you want to use this, say you're a primatologist, you can be on the primates feed. Each feed has its own rules, but you have to show that you're actually a primatologist. You can't use it to spam about world news. It's only primatology. So it's a very, very useful tool for scholars. And you can also follow people in big groups. So there's a bunch of starter packs on there. In fact, one of the organizers, Vivek, has one of his own. And I've listed a whole bunch there. So by the end of this conference, you could easily follow a few thousand people, have your own nice little ecosystem of animal behavior researchers. And just a little reminder, that's a Blue Sky thing, which is always to make sure you use alt text to keep it accessible for everyone. So I'm really excited to see everyone's posts. I would really encourage everyone to sign up today. You can find us at asab.org. And again, I'll have some of these little tips and tricks to make Blue Sky more fun. But really, it's very simple, and I think we're all going to enjoy it. So thank you.
Well, hi, everyone. So we're going to start with my name is Cedric. Hi. We're going to start with our first plenary speaker, which is Malin Hacking. Malin is associate lecturer, associate professor in gender studies at the Örebro University in Sweden. She came all the way from there by train. It took her 32 hours over 2 days. So she's very happy to talk to you afterwards about a train journey in Europe. She also confessed that she's, as a fun fact, she's also doing Kung Fu. So I don't know if that makes her more approachable. As Joe mentioned, she's going to give the public outreach lecture, and we have two classes of high school in the front row. from the Royal Art School, Edinburgh, and the Edinburgh College. Malin is an evolutionary biologist and a gender researcher, and she uses her expertise in those two fields to teach gender and queer perspective on biology and study the controversies in sex difference and how they emerged. Malin graduated from her PhD in evolutionary ecology at Stockholm University in Sweden, and then she did a postdoc at the Center for Gender Research at Uppsala University. From there, she conducted an overview on variation in mate choice among animals at the University of California, Los Angeles. She examined the gender biases in genital evolution research at Macquarie University in Australia. She held courses on gender and queer perspective in different universities in Germany and Sweden. Malin is otherwise known for her work in variation in sex and sexuality in animals, and for studies of sexual selection research and why females are understudied. And more notably, she's known for a book, The Female Turn: How Evolutionary Science Shifts Perception about Females, which is also the title of her talk today, and what she's gonna tell us. So, Malin, welcome.
Thank you. Thank you so much for inviting me to have this talk. This is kind of a dream conference for me to come to. So I hope everyone feels that I'm approachable. I want to talk to every one of you, and especially welcome to the college students. And thanks for inviting me to having this talk. And I should also mention that I used to do Kung Fu. It was a long time ago, so maybe that makes me more approachable. I'm going to talk about this book and this study I made about the history of how perceptions about females have shifted in evolutionary biology. So I should first just tell you that I started out as an evolutionary biologist studying animal behavior, and it was my PhD in zoology. It took me about 5 years of studies, research studies. And after that, I have become an associate professor in gender studies, which takes about 5 more years. And this is a title that makes it possible for you to be an advisor for PhD students. So this is kind of a path in the career of researchers. So this means I'm both an insider and outsider of the field of evolutionary biology that I'm studying. So my starting point in doing this study is a feminist critique of natural sciences. It's not a apolitical, value-neutral, and objective process. Instead, who's doing science actually affects the kind of knowledge that we are producing. And the history of science has shown this, right? So, for example, about 100 years ago, it was a medical theory and something that medical researchers believed in, that women had a limited amount of energy. They could only use their energy, or if they used too much energy for their brains, like intellectual work or studying, then their most important role in society would be threatened because their ovaries would shrink. So they didn't have enough energy to both do the intellectual work and reproduce. So that was one reason why women weren't allowed to universities. And this is true. So this is one way in which we can understand how knowledge, and in this case medical knowledge, was formed by the male-dominated then. Women weren't allowed, so the male-dominated science. And so there has been a lot of feminist critique against the male focus in science. And we should also understand our science as a cultural and social process, because even if any one of you make a discovery, It doesn't become knowledge in our community until we have persuaded the community that this is something that is actually knowledge. So it's a social process. It's a community that produces this knowledge. It's not us, our individuals on our own. We have to make the community understand that this is actually something of a discovery. So, and also this feminist critique and the feminist researchers have described how researchers' ways of seeing form their specific knowledges. And we cannot do the God trick of knowing everything or seeing everything from nowhere. We have to describe how our particularity, our specific perspective produces the science that and the knowledge that we do. So my study case then, what I wanted to understand was how perceptions had shifted about females in evolutionary biology and animal behavior. So we know that Darwin described, and traditionally females have been assumed to be coy and passive and mating with only one male. But this has shifted towards acknowledging that females can have active strategies and frequently mate with multiple males. So how and why have these perceptions changed in the community of evolutionary biologists? So what I did was decide that I wanted to talk to the pioneers. Of course, I read a lot of the scientific literature, but I also wanted to interview researchers. So I wanted to interview the pioneers who had presented female-centered hypotheses. I also wanted to talk to people who are very well known in the field, so very highly cited researchers in sexual selection. And the third group was the feminist researchers within evolutionary biology. And these are not strict categories, they're kind of overlapping as well. So here are the ones I interviewed. And I also used a theoretical framework to try to understand these shifting perceptions, and that is the production of ignorance. And it means that studying how knowledge is ignored or delayed or forgotten. And a famous science historian says that ignorance is not just that we don't know, it is often the outcome of cultural and political struggle. That's Londa Schiebinger who says that. And there's this wonderful study by Nancy Tuana in 2004, in which he describes the knowledge about the clitoris and how it has been known and become unknown in different times over the centuries. And one example of this is this picture of the genital tract of a woman. It's published in a textbook in the 1966. And you can't see any sign of a clitoris in there. It was just not interesting. And then you see this picture from the women's health centers who were really engaged in trying to understand the clitoris, trying to make people have more enjoyable sex. And there you can see both the clitoris in its erect version and its non-erect version. So this is one example of the knowledge, how kind of our understanding and our knowledge about something depends on who is doing the research and what kind of interests is in this community of researchers doing the research. So what I brought with me with this perspective is how did the researchers' specific ways of seeing form their understanding of females, and also in which ways have ignorance been produced in this field. So everything starts in the field starts with Charles Darwin's theory about sex differences. So when he had presented the theory of natural selection, there were still some things that were troubling, and he got a lot of criticism for certain kind of traits, for example, these male plumages, that couldn't be explained by better survival of those individuals that have them. They would rather be bad for survival. I mean, if you're really beautiful plumages, you could be eaten easier than if you didn't have them. So he presented the theory about sexual selection, that these plumages, for example, could have evolved by females choosing males who had more beautiful plumages. And he also described women, in line with his society, as morally superior to men, but intellectually inferior. If you read the book, The Descent of Man and the Sexual Selection in Relation to Sex, you will see that Darwin described many, many species and the sex differences in all those species. But when he then generalized, he also described it in rather stereotypical terms, like the male is a more active part in the courtship. The female, on the other hand, with rare exceptions, is less eager than the male. She is coy and may often be seen for a long time endeavoring to escape from the male. So, many have criticized Darwin for bringing in those kind of stereotypical Victorian notions into the theory of sexual selection. But it's not only that he formed his theory in line with contemporary Victorian ideas, he also did it against, in certain ways, the Victorian ideals. And this is something we've learned from Evelyn Richards, who has written this wonderful book about how Darwin developed his ideas about sex differences. So, for example, he, in this time when the woman question was debated a lot in society, women wanted to study at universities, and women wanted rights to vote, for example. He argued against John Stuart Mill, and John Stuart Mill was a philosopher who described women's. He said that if women only could be educated, they could also become geniuses. They could also do intellectual work. But Darwin argued against him and said that this is how humans have evolved. Men have competed among each other more. Men have fought more for survival. Therefore, they have also developed these intellectual traits that women don't have. So this was in line with the Victorian ideals then. But against Victorian ideals, he described female choice as a very important factor for the evolution of male traits. So in this way, females were actively making males develop these traits. And this was also an idea that his contemporary biologists didn't really believe in, so this idea didn't get a lot of attention in the research until much later. But what Evelyn Richards says is also that Darwin's view on women it wasn't that he was only, sexist. It was a logical effect of how he thought about inheritance and how embryos evolve and develop. And also his personal experiences did not talk against this. He was surrounded by nurturing and caring women. And in a part of this book, Evelyn Richards says that it doesn't seem to have occurred to him that one reason why these women were so nurturing and caring was his being sick and making them care of him. So what I found was, and other people have also written about this, that there was an early female turn in primatology, in the research on primates and monkeys. This is Sarah Bluffer-Hurdy. She is one of the pioneering feminist researchers. She went to India to study langurs, and she wanted to investigate why males often killed young infants in these langurs. So the predominant idea at the time was that there was some kind of social unrest, and in those times of social unrest, there was some killing. So this was a kind of sickly behavior. But when Sarah came there, she realized that it's often the case when a group of males takes over a group of females that they kill the youngest infants that hadn't been weaned. And she thought maybe if they did that, that could actually be a behavior that had been sexually selected. So in line with Darwin's theory then, that if they kill off the offspring, maybe they can get more offspring themselves, because those females will get children, their own children, earlier. So she proposed that this was a sexually selected strategy from the male side. But then she also felt a lot of empathy towards the females. So what would you do? Why didn't the females do anything when their infants got killed every other year? So she started to ask questions that her male colleagues hadn't asked before. So do they have any strategies? What do they do in order not to have their young killed? And she followed them in the field, and she found out that the females often sneak away from their group and have sex with initiate sex with many other males around. And she proposes that this is a counter strategy for those male strategies, because if the males don't know who is the father of the young, and if those males then take over the group, then that could prevent their children from being killed, their infants from being killed. And she has also been very vocal in criticizing stereotypes in sexual selection theory, like the coy females. She didn't find her females to be coy. And she also wrote a book in 1981 called The Woman That Never Evolved, and it was a criticism towards this theory and the male focus in the theory. So she was one of there are several pioneering feminist researchers in primatology who were important for shifting perceptions about females in primatology towards including aggressive females and competing females and sexually active females. But there's also an interesting parallel history of the primatology in Japan. It developed independently from Western primatology, and it brings with it a lot of cultural notions from Japanese culture in it. So in Japan, there's not this disruption between humans and animals. It's more of a, where both humans and animals have agency, and they were trying to understand the species society. How do the relationships work in this society? So they were really early on realizing that females have power relationships, and the kinship relationships are really important for the power relationship in these macaques and in many other monkey species. So they actually realized that much earlier than the Western primatologists, who had these perceptions of females being passive. And in bird research, the change in perceptions about females came later than in primatology. So birds was and is still a classical animal group for studying sexual selection. Darwin wrote a lot about birds, and it's also a group that brings a lot of questions about the evolution of sex differences. And in this group, since 90% of birds are socially monogamous, the females were also assumed to be monogamous. But DNA fingerprinting, the same technique that we use to test whether our social father is our father, has started to be used in bird research. And this revealed an enormous amount of multiple mating, that is, that females mated with several males, much more than we had assumed before. And this led to what some call a revolution, and because these supposedly monogamous females then were shown to be mating with multiple males. But the early explanations for this were very male-focused and focused on male strategies, saying that it's not what the females wanted, it's more that the males were imposing those extra matings with other males. However, there were bird researchers who questioned these interpretations. So Patricia Guati presented one of the first studies in which females were shown to mate with males outside of their social pair. And when she presented this at the conference, there was a famous ecologist, Steve Amnon, who said to her, "Patty, those females were raped." But Patty didn't think that those were raped. She thought that, you know, it's really hard for someone to rape a passerine because these birds don't have penises. Maybe you don't know that most birds don't have penises. So these small birds need to put their cloaca towards the cloaca and transfer the sperm from the male to the female. And in order to do that, the female almost had to help out, or she could just fly away. So within the community of researchers, there were this predominant explanation that males were the ones imposing these extra pair matings. And there were also some people who did not believe in that explanation, like Patricia Guati. And she is a pioneer feminist ornithologist, and she says that I really am interested in female biology. I'm a female. I would like to know more about that. So this is one example of how our perceptions or our perspective influence what kind of knowledge we produce. And a later study by Susan Smith, who's also a feminist bird researcher, she tested these assumptions about passive females that she didn't really believe in. So if males were imposing those matings onto females, the females would just be passive, sitting in their territories and mating with whomever is coming up in their territory. And she didn't believe that, so she put radio trackers onto the females of these black-capped chickadees, and she followed them in the field. And she showed that the females actually sneak out in dawn and seek extra pair copulations in territories with males of a higher rank than their social mate. So this was a start of shifting perceptions towards females actually can have sexual active strategies in birds. So this is one example of a pattern that I've found in this history, that we often focus on male-centric investigations first. The research starts with male-centric investigations or explanations, and thereafter it includes female-centric equivalents, and this is a common pattern in our field. So when I interviewed an insect researcher, Daryl Gwynn in Toronto, he said, well, we already knew, as insect researcher, we already knew that females were mating with multiple males. Because in the 1950s, we started using something called the sterile male technique. And this was used in agriculture, in pesticide. So in pest, when they tried to get control over pest insects. So if you take a male and you take radiation and all his sperm dies, and then you can spread out a lot of those males in the field. So, and this works as a way of reducing the population of pests, but it only works if the females are mating with only one male. If there is a species in which females are mating with several males, then it doesn't matter if she mated with one male who was sterile. So since the 1950s, insect researchers had known about many species in which females were mating with multiple males. And this was actually the basis for a very interesting new theory, the sperm competition theory. So Jeff Parker at Liverpool, he suggested that not only as Darwin described the struggle for mates and before mating, we could extend this theory to also happen after mating. So not only males compete among each other, but also their sperm can compete among each other. And when you extend a theory like that, you might have thought about, both the male part of the explanation and the theory and the female part, but Parker was very focused on the male part, the sperm competition. And he worked on yellow dung flies. He told me that he used to play with a friend at a farm when he was young, and there were yellow dung flies there, and you could see how they competed among each other, how they fought. And when he was then suggested several different species, he chose the yellow dung flies because he knew them and he thought it was interesting. And he also says that it was easier to look at the males, to focus questions on males, and perhaps due to his male perspective. He also did observe females and wrote about females in his thesis and the papers coming out of his thesis. But he often describes them as, interpret them as very passive. And he even describes in his field research that he went out and lay down in the field beside a cow dung, watched these yellow dung flies, and sometimes it happens that a female actually lands before a male, and she does a special dance, like having her wings like this and then like this. But he never describes this as some kind of active choice. But I would interpret that as this is maybe in this system, in this species, which is very much dominated by male-male competition. Males are pushing off smaller males when they're mating with a female and then take over the mating. So this is maybe the only chance that a female has to do some kind of active choice, doing this dancing in front of the female. But he never describes this as that. But this male focus is also in line with this sexual selection theory. And his dung fly system, the species he's working with, is very much, as I said, dominated by this male-male competition. And this dung fly system is also what has inspired him to think about a lot of many other theories. So here we see how the species someone is working with is actually forming very much of their particular knowledges. So the theory of the female part of competition after mating is called cryptic female choice. And this term was coined by Randy Thornhill in 1983. And by cryptic female choice, we mean that females can actually control which sperm fertilize the eggs. And I thought when I went to interview Randy Thornhill, I thought it was a little bit of a feminist paradox. So how come somebody who has written the book, The Natural History of Rape, which some of you may know and some may not know about, but he drew parallels between what he called rape in insects and rape in humans, and said that this might be a sexually selected strategy also in humans. So we should actually teach men that they are potential rapists, and we should actually tell them this and make them not to be rapists. And this book was very much criticized from a feminist perspective and from other perspectives as well, social science perspectives, when it came out in 2000. So to me, it was a bit of a paradox that he was the one who had presented a very female-centric hypothesis. He describes in his papers, he describes females as very active, they avoid mating with males who don't give them the gifts. So his study species were hanging flies, and here you see a scorpion fly. But he also describes, so he describes what he calls rape, but he also describes how the females manage to break free from males that attempt to copulate by force, and when they are forced, they become sexually receptive quicker and somehow cause low insemination rate. So the females can somehow influence how much of those sperm she's using. But when I had talked to Randy Thornhill, I realized that this is not so much of a feminist paradox, because his perspective is purely biological. So he observes these insects, and he sees that the males are forcing the females, but he also can see that the females are actively working against this and in different ways. I also found that there was this shift in snake research. Jesus Rivas told me that he was the first one who described the snake mating system as polyandrous, so that the females mate with several males. But at the time, in the 1990s, when he started his studies, all the snake mating systems had been described as polygynous in some way, so that females are mating with several, the males are mating with several females. But when he went back and looked into how other people had described other species, he found several species which he would describe as mating with several, females mating with several males as well. So he kind of did a revision of the description of the classification of mating systems, in which the part in which females had mated with several males had been ignored before. And I also decided to interview a spider researcher, Marie Herberstein, because it's kind of interesting with spiders. That research has been very much focused on females because of their ecology. So the females are often much bigger, they are staying in the same place, while the males are smaller, they get eaten by the females, and so it's much harder to study the males. So the whole spider research has often focused much more on the female side. And she said she didn't find much of gender bias in the reception of her research on spiders. However, she found a kind of bias that it is a taxonomic bias, so that it feels like people in the research field, in the community, didn't think her research were as important as bird research, for example, because spiders are just, these kind of little, not ordinary kind of animals. So many people are thinking of them as exceptions in some way. And this is also what many other researchers find, that there is, and it is acknowledged within the community of animal behaviorists, that there is this kind of hierarchy, that within sexual selection research, we often cite the bird research much more than the spider research. And bird researchers much more cite among themselves, while everyone else working with fishes or anything else need to cite the bird research. So there's a kind of citation hierarchy. So to summarize the production of ignorance about females in sexual selection, I found this repeated pattern of male-centric investigations first, and then turning to the female part. There's also loss of acquired knowledge, because really early on in the 1930s and 40s, I can find studies in which the researchers actually describe the females as mating with several males and being active in the mating. But this hasn't influenced the kind of predominant view of females as passive within the communities in this bird research and primate research at the time. I've also found that there is this undermining of the authority of certain knowers. For example, the Japanese primatologists' research, when it became known to the Western primatologists, many of the Western primatologists were very skeptical about the methodology, and they didn't cite these research findings because they didn't believe in the authority of the Japanese researchers and their methods. And production of ignorance also happens through this widely acknowledged hierarchy. And to summarize how the researchers' ways of seeing have influenced the research and the knowledge we have produced, it has been as feminist, as engaged in female perspectives, due to particular methodologies, due to the knowledge gained from other research fields, like the pest control studies, and also the cultural context, that the Japanese versus the Western primatology, and the influence of the study animals, which was important, I found. And this has made certain researchers particularly engaged in forwarding females as active, aggressive, and variable among themselves. However, as I said in the beginning, a discovery doesn't become knowledge until it has been critically recepted in the community of knowers. That's Helen Longino, feminist philosopher, who says. And so it wasn't until we had this social and political context of the feminist movement and the sexual revolution that this female turn has shifted. And if we talk about gender in science, we know that historically the professionalization of science excluded or marginalized women. They weren't allowed to be researchers. And the progress of the feminist movement have enabled women to take part and do science. And in this study, at least, both men and women challenged the assumptions about females. And there wasn't a direct relation between the gender of the researcher and being an initiator or proponent of female-centered hypotheses. But those who describe themselves as feminist scientists often worked strongly to identify gender bias and express an outspoken strategy to investigate female animals. And in line with this, talking about how we can acknowledge how our perceptions influence our knowledge, my own knowledge about this female turn, which I've described, is also partial and dependent on my ways of seeing. So it's dependent on my point of departure, my experiences as working in the field of animal behavior and evolution, my knowledge about the methodology, my network of researchers that I know, and also all of these gender analysis that I've made on the field. This has formed my research question about the females. One could also study how perceptions about males have shifted, because they have also shifted. But I was interested in trying to understand how the perceptions about females had shifted. And also my position may influence the kind of material that I get to work with, my relationship with my interviewees. Perhaps some interviewees did not want to talk to me or talk to me in a certain way because I'm a feminist biologist and both biologist and in gender studies. And also my interpretations are influenced by my analytical tools. I choose to look at the production of ignorance and this how different particular researchers understand and how their knowledge is formed by their experiences and their theoretical perspectives. So this has led to this writing the history of the fields from a gender perspective. But we still have continued ignorance about females. This female turn is still an ongoing process. And I think the knowledge about how females, how sexual selection works in females is much less studied than in males. So this is from my paper in Nature Communications, in which I look at the number of publications which looks at competition between males, sexual selection in males. Those studies are much more numerous than in females, and that continues to be the case today. There are different reasons for that. Both there are kind of practical reasons. It's much more harder to look at female sexual selection, but there's also because of these different traits that may be much more common in males, like the bright plumages, for example. So there are biological reasons, there are practical reasons, but there's also this gender bias and our biases in the theoretical perspective and what we think about. And I also think that, you know, how we define what is sexual selection influences this, because often how we define sexual selection as male, as competition for mates and fertilizations, actually excludes a lot of ways in which females compete among each other. So females compete among each other for reproduction in other ways than competition for mates or fertilizations. Sometimes they lay eggs in other females' nests, and that's not called sexual selection. So this is also one reason why it looks like this. But we think we need to bring this with us when we do these kind of studies and think about whether our biases, whether theoretical or otherwise, influence the research and the knowledge that we produce. So, thank you.
Yes, good morning, everybody. It's my pleasure to kickstart this session. I just have to warn the organizers that my way to deal with biases in animal behavior is to embrace the biases. I love biases, so today I will have a chance to tell you why this is the case, starting from the beginning of life. So, yes, last year I told you a story about some biases that the baby chicks soon after hatching have. They have a preference for objects that instead of going down, following gravity, go upward against gravity. So they have some biases, some expectations for the physics of motion. I'm not the first to do these studies. You might be aware of the preference for biological motions that not only baby chicks, but also infants have from the beginning of life. So the semi-rigid motion, biological motion, versus the rigid motion is preferred. So we have a bias for this. But there are not just biases for approaching objects. There are also biases to go away from objects, not only to behave in a different way when there is a looming object that approaches and has an increase on the retina compared to sweeping objects. So the chicks here quickly goes away from looming objects, but when there is instead a sweeping stimulus, rests, kind of freezing, because that can, instead of being a predator that is approaching quickly, can be a raptor, for instance, that is patrolling and checking the territory. So this is another bias for avoiding some stimuli. But motion is very attractive. So from the beginning of life, we also have a preference for objects that instead of having a linear motion, they change in speed. This is not just for chicks, but also for human babies. So this was discovered a few years ago, and we repeated this in 3 breeds of chicks, finding that, in fact, this is the case. But this happens in day 1. 2 days later, this preference has disappeared. So the biases are widespread, but first, not all the animals respond stereotypically in a fixed way, and then they are plastic, they are transient. Okay, so it's why. Let me tell you something about different biases, so patterns, so static patterns, like the face-like pattern, 2 blobs on the top and 1 at the bottom. Yes, not only chicks, but also human babies have this preference, and we wanted to know whether there is a widespread bias also for solitary species, like tortoise hatchings, that don't have parental care. And so we hatched the tortoises. Let me see if the video goes on. Yes. And there are the baby tortoises. They haven't seen any face before. And we give different patterns, and they consistently go towards the face-like pattern. So again, another bias from the beginning of life, even for static patterns. And maybe we wonder whether the chicks that hatch usually with the siblings, they have some biases that can help them to then imprint, to orient and then imprint towards the mother hen and adult compared to the siblings, because it would be very disadvantaged, right, if they imprinted on the siblings that know so little about the environment. And so we reasoned that a very salient cue for distinguishing the adults from the siblings is the size. And then with Vera, that is here, and other researchers, we measured the color of the feathers, and we discovered that the chicks are lighter and the hens are slightly more reddish than the chicks. And with a series of experiments, we found that indeed, not only chicks prefer the reddish colors, but there is a stronger preference if these are, the red color is big. And so adding cumulatively multiple features has more than an additive effect. And then the classic biases for colors. They have a preference for red versus yellow, without having seen these colors before, right? They have a preference for blue versus green, blue versus turquoise. So a lot of biases. So why are we born with these biases? One reason is that it is advantageous to have an orienting response towards these stimuli that are salient, associated with living beings. But I wonder whether there is something of biases that help us also in learning. And so, with the same colors, we expose the chicks to just one of these, either blue or red, green, yellow, orange, or turquoise, and we look then at the generalization learning curve for the imprinting color versus other color on the continuum. And strikingly, for the red and blue, we found, as you see here in this part of the plot, we found a gradient, a generalization gradient learning. But for the other colors, not. And this is interesting. Even with the turquoise, they stick with the preference for blue. So what we find here is that the initial preferences have an influence not just in the moment, but also for learning. And strikingly, these colors, blue and red, are those that are rare in the environment. So they are good predictors of the presence of something that is not the background, but that it is an interesting piece of information. For instance, the mom. So learning, priors, biases, support learning. However, we are still here with the question of why these biases are so soft if they are advantageous. You see here that there is a lot of variability. Some chicks go on the opposite side. So why, if they are advantageous and widespread, why they are so soft? Why they are transient? So we can think about a model that puts all this together. On the one hand, we have the additive model. On the other hand, we will have a combinatorial. So for the additive model, we have multiple features. And you see here the black bar. On the right, there is attraction. On the left, there is withdrawal. If we have optimal features, we can add attraction for each of them, and we sum. And if the chicks are exposed for a longer time, there is more statistical evidence, and they can like more and be more attracted. They are learning. The reverse if there is aversion, right? The more cues are aversive, the less they will be attracted. However, this doesn't correspond to what we observe, where there is an advantage in multiple cues that is not additive. So probably what happens here is that we need to have soft biases, because if we base our choices just on one cue, it's very easy to make a mistake. But when there are multiple cues, there is a combinatorial advantage, so that in the same time we can very quickly multiply by the presence of the occurrence of multiple features and make adaptive choices. Learning is hence very sustained by biases. Let me show you another study that we ran with chicks, again, hatched in darkness, and exposed tactilely to either a smooth cube or a bumpy cube. So they didn't see anything, but they could touch either the smooth or the bumpy cube in darkness. And then the first visual experience arrived. The chicks were exposed for the first time to light, and they could not touch, but they could just see the smooth versus bumpy object. And what did they do? So you see here a chick that was exposed to the bumpy stimulus. And so first visual experience, looks around, it's the first experience, so let's wait a bit because they have to learn basically how to move in the proper way. And then the chicks approaches the bumpy stimulus. This is just one video, but look on the right at the plot. The chicks that had been previously exposed tactilely to bumpy stimulus go more than the smooth chicks to the bumpy, and vice versa. So it seems that we are hardwired from the beginning. We don't need experience to be biased and match tactile and visual information. To wrap up, you have seen why I like biases. They have an adaptive role in the initial orienting responses, changing behavior by local enhancement, a combinatorial advantage of soft and plastic predispositions, enhancing learning, generalization, and induction.
Thank you. Hi, my name is Arzo. I'm an undergraduate student from Stockholm University, and today I'm excited to share my research with you, where mate choice copying is often studied with a strong focus on females, under the assumptions that males are less likely to engage in this behavior. But is that really the case? In this talk, I want to rethink these sex bias assumptions and explore whether males might be more willing to copy mate choices than we currently expect. So to understand mate choice copying, we first need to revisit the roots of sexual selection. Darwin identified two key mechanisms of sexual selection, where one of them is male-male competition, where we typically see males fighting to compete for access to a mate through agonistic interactions or displays of dominance. And then on the other side, we have a female choice, where we typically find females choosing mates based on traits like health, genetic fitness, or parental abilities. But mate choice isn't always this straightforward, and these roles in sexual selection are not always this clear-cut, with females always being the choosers. What about female-female competition and male choice? Well, mate choice is complex. Individuals seek high-quality mates, which display beneficial traits to pass on to their offspring, but also a partner that won't just neglect the kids, like having great parental care. However, evaluating every potential mate in a group is costly. It takes time and increases exposures to risks. Instead, individuals can rely on social information, a phenomenon where observing others' choices acts as a shortcut for their own decision-making. And this behavior applies to mate choice too, known as mate choice copying, which helps reduce uncertainty in mate selection. But as indicated, we typically discuss this behavior in relation to females. This reflects a bias in the field, as females are often assumed to engage in such behaviors due to their higher investment in reproduction. But what about males? Could they also use social information for their mate choices? So according to this research by Alice D. Davis and others, where they conducted a meta-analysis of factors influencing the strength of mate choice copying, something surprising was revealed. There was no difference between males and females in the tendency to copy, which is unexpected, because if you think about it, if males who copy, they may substantially be increasing their exposure to sperm competition, mentioned earlier, a significant risk. So why would they do it? Well, there's a possible advantage here. Copying could save time, allowing males to focus on females that already display signs of receptivity, especially in group living situations where females are receptive at different times. But I also want to highlight that the meta-analysis also displayed a significant disparity between the sexes tested for mate choice copying, where we have the proportion of effect sizes being 91% on females compared to 9% on males, with a clear underrepresentation of the males aspect of this behavior, suggesting the need for more empirical work. And that's where our research comes in. So we designed an experiment to test mate choice copying in males, focusing on a non-traditional model system that meets specific criteria. So let me introduce to you the pygmy half-beak. What makes them so special for this study? Well, pygmy half-beaks are live-bearing fish native to Southeast Asia. They naturally live in bigger mixed-sex shoals, ranging from 6 to 128 individuals. This is a species that are specialized surface-dwelling fish. They spend most of their time within the first 2 centimeters of the water column in lakes and streams to feed on insects. It is typical to see females being surrounded by males, as you can see here on their, from a picture, from their natural habitat, where one female is closely surrounded by 3 following males. So this is what a courtship of a pygmy half-beak looks like. When it's time to reproduce, a male simply goes underneath the female, and they start courting the female by chattering their jaws, and the female shows that they're receptive by staying absolutely still. And there's the mating, lasting for about 40 milliseconds. So it's very quick. So we're gonna show this in slow motion too. So they go underneath, they chatter their jaws, and that's it. There's the mating. So they have a clear courtship behavior. So, but let's look a little bit closer to what's actually going on here. Females phenotypically advertise receptivity through an orange-colored ornament on their ventral side of their body called a gravid spot. Typically, half-beak males use this ornament as a mate selection cue, positioning themselves closely beneath a female, attentively evaluating this trait. And it is suggested that males assess female receptivity based on the size of this ornament. So this is a study done by Ogden and Fitzpatrick, where they looked at female brood cycles. They give birth every 30 days, and as you can see there, the gravid spots get bigger with time, and then they give birth, and then it shrinks. And we interpret this as a physical pull of the skin around the female, as when her embryos are developing, that's pulling the gravid spot wider and wider. And males also have a preference for this large gravid spot, where they spend a significant longer duration for courtship behavior. And what this could mean is that males might want their sperm first in line to fertilize the next clutch of eggs. So we see a lot of phenotypic variation in female quality. And here is an additional cool picture. You can actually see unborn offspring inside the female through their semi-transparent skin. So this might be a very interesting female for the males. But for our setup, our experiment was conducted in a laboratory setting using a dichotomous choice assay to test male preference. One of the stimuli was always a pair, one female and one male. By contrast, the stimuli presented in the other compartment varied and are referred to as treatments. And we applied association zones to each side of the arena to track the focal individual and quantify the duration it spent in each association zone. It's the 5 centimeter zones there. And if fish were acclimated to the arenas without being able to observe each other, and then we removed opaque barriers and let the games begin, meaning they were now able to visually assess each other through transparent barriers and engage in olfactory detection. We recorded and tracked a total of 91 focal males. So for our first treatment, we tested male preference for a pair over one female, and this is what the results showed. On the x-axis, we have the given treatment, and on the y-axis showing strength of preference. Everything below 0 represents no mate choice copying, and everything above 0 represents mate choice copying. And our findings suggested a marginally non-significant trend towards the pair. This could indicate interest in receptive females, signaled by the presence of a male, though it's also possible that they were just attracted to the larger group size. But we also quantified courtship behavior in the pair using behavioral observation research interactive software, also known as BORIS. Specifically, we counted the duration of courtship displays performed by the pair male. So when testing if the amount of courtship behavior performed in the pair predicted the preference of focal males, we found a significant positive correlation between courtship duration and strength of preference, telling us that duration of courtship displays seemed more interesting for the focal male. So for a second treatment, we controlled for group size by offering males a choice between a pair and 2 females. And surprisingly, males did not show a strong preference for either side. Even with the choice of twice the reproductive opportunities, we still see some individuals choosing the pair. However, this raised questions about the role of sex differentiation in their behavior. So lastly, we replaced treatment females with males, instead to test if the observed behavior were due to sex differentiation or group dynamics. And our findings shows that males showed a strong preference for the pairs, indicating they could differentiate between the sexes and were not merely shoaling with males. This suggested a genuine interest in females. So to summarize this, in treatment 1, there was a trend towards the pair, suggesting potential interest in receptive females. Treatment 2 showed no strong preference, though some males still chose the pair. Treatment 3 and 4 showed a significant preference for pairs, confirming males can distinguish between the sexes and are drawn to females. So as our results show, we see trends linked with male mate-choice copying, indicating that something's going on with the male's preference, particularly with the BORIS measurements and the correlations. It appears that context plays a significant role in our experiments in determining mate-choice copying. So this research elevates the discrepancy in research concerning mate-choice copying, encouraging us to rethink these traditional assumptions that males are less selective or don't engage in mate-choice copying. By conducting further studies similar to this one, we can better address this bias and advance our understanding of male mate-choice behavior. And I would also like to thank all these amazing people for helping me, but a special thanks to Leonard Winkler, my colleague, and my mentor and supervisor, John Fitzpatrick. And thank you for listening.
Okay, welcome back, everybody. Good afternoon and welcome to the afternoon session. My name is Amy Leedale, and I have great pleasure in introducing our second plenary speaker this afternoon, Antisa Tulina. Antisa is an evolutionary ecologist by training. They did their PhD at Oxford University, studying the formation and dissolution of pair bonds and life history trade-offs in monogamous birds. Since then, Antisa's work has focused on meta-science and open science research. First, as a researcher at the Netherlands Institute of Ecology, and now as a senior researcher at the Ruder Bošković Institute in Croatia. Among several brilliant open science initiatives, Antisa co-founded the Spy Birds Network and Database, which brings together researchers studying populations of individually marked birds, and the Society for Open, Reliable and Transparent Ecology and Evolutionary Biology, or SORTI. Outside of academia, Antisa is also very musical. She plays the piano and has also taught the piano as well at her time in Oxford. And a fun fact about Antisa is that she was also a lead singer in a band called the Balkan Wanderers in her time at Oxford University. Today, Antisa is going to talk to us about using open science tools and practices to work towards a more robust and reliable research in her talk today titled Do Incredible Science, Why Would You Care? So please join me in welcoming Antisa Tulina.
Thank you for the introduction. I'm sorry, we have a lot of sh, ch, and similar letters in our names. And it's very nice to be here today because I think my first conference for PhD, so where I presented something related to my PhD, was ASAB conference. And I presented a poster on the approach I wanted to use to study dissolution of pair bonds in birds. And since I haven't been to ASAB meetings, so I'm really happy to be here again and listen to all the talks and looking forward to all the rest because it's really interesting. And I tried to find a common line in my research because I changed a lot. So I didn't have this like line where you just research something and then you go and learn more about it. I kind of started one thing and then that led me to something completely new and then I started with that and then with something else. And I tried to find this common thing and I think it was always something related to informed decision making. First related to animal behavior and ecology, where I studied formation and breakup of pair bonds, so how animals do this and what are the consequences of these choices. And also I worked on life history trade-offs and site selection in bats. And then also informed decision making by scientists, but also by society at large. First, as actually conservation researcher, even before my PhD, where I worked for an NGO, mostly on birds and protected areas in Croatia. But then later, in a more scientific approach of how to deal with uncertainty in the information that we collect. Also, I do a lot of research synthesis. So you have a lot of information on a certain topic, and then you try to synthesize what we actually know. And recently, I'm really interested in the scientific process. So how do we do our science? And does that lead to correct and robust knowledge? And this is me when I was still in Croatia, touching a lot of animals. That's what I liked. And I was at a conference somewhere, some bird conference. And somebody from BirdLife gave a talk on the bias in research of birds. So the most studied species with the most publications was great tit. And the one with the least publications was, well, I don't know the English name, it's Sita neumeier. So we have this European nuthatch in Europe, but there is also this one that has more eastern distribution. And Croatia is actually the western part, so it's like the end of the range of this bird, and they do display some specific behaviors there. So I said, well, for a PhD, I'll work on Sita neumeier because it's not well studied, but I ended up working on great tit, which is the most studied. And this is also one of the biases, because I think especially for a PhD, you don't want to go into something completely unknown and start from scratch, but maybe work on something that's kind of safe. And that's what I've done. And I don't regret, but maybe I once worked on Sita neumeier. And when I was doing all of this, I really thought that science is systematic and objective, and it's evidence-based, and it's unbiased, and not dependent on cognitive biases. But then, during my PhD, I changed my opinion. I still think science is great, and is one of the best ways to understand the world around us. But also, I encountered some issues when I was doing my first meta-analysis. And for that, I had to read a lot of papers, but very in depth. So comparing things and thinking about the process, analysis they've done. And what I found is that they were not very well written. For example, values would not match. The one that was in a graph was different from the one reported in methods. There was a lot of missing information, so it was unclear what was the sample size, something very simple. And methods were often unclear. So I still wanted to use these studies in my meta-analysis, and I tried to contact the authors to get to the data so they can tell me what was the sample size. And I sent a bunch of emails, many have never replied. And often when I did get reply, I would be told that they didn't know where the data was. And this struck me because sometimes it was really long-term data sets of 30 years that, there is money and time and expertise invested into collecting these data, and then you have few publications out of that, and then they are forgotten somewhere, lost. Nobody can ever use them again. And obviously I also learned the publication bias, so that some studies, types of results are more likely to be published than the others. And this is mostly those that are statistically significant or somehow interesting. So in my talk, I will tell you a bit about the issues that we have in scientific process, then some solutions to these issues, what are the challenges in achieving or applying these solutions, and I will talk about these two things, the second and third, somehow at the same time. And finally, what can you do to make your science more robust? Okay, let's start with issues. I'll talk about publication bias because I think most of you have heard of that. It's been described for a long time, but it's still present. And you can say, well, who cares? Because if we really published everything, all the boring findings, wouldn't we just clutter literature with something that's not really interesting? So let me take you through a little experiment where we think that there is 50% chance that there is effect that we try to detect. We set alpha to 0.05. That means if a null hypothesis is true, then false positive will be get in 1 in 20 trials. So just because how statistics work, you always sometimes get false positives. And we set power to 0.8. That means if there is effect, we will see it in 8 of 10 trials. That still means that in 2 trials we won't detect it. And power, amongst other things, also depends on your sample size. So if you have higher sample size, you're more likely to detect an effect. And actually in ecology in general, also I guess in behavioral sciences, power is usually much lower than 0.8. We don't have such big sample sizes. And let's say that we have 5 published studies where we have 3 that showed green, so it's positive result, and 2 show that there is no effect. So I ask you now, who thinks that based on this, so 3 positive, 2 negative, that the effect very likely does not exist? Who thinks that? Nobody, okay. Who think it could exist, but it's unclear still? Okay, that would be my take too. And who thinks it very likely does exist? You're right. Based on something that's called, I'm not going into that, but it's called log odds ratio of true effects. Basically, it gives us some kind of, well, trust in our effect. So the first study, that first dot increased our trust, then the second one decreased it, third one increased it, and so on and so forth. So it's above 4, so we believe there is an effect. But if I tell you there were more trials, and you see this, I think I don't have to ask you that most of you would say effect very likely does not exist. And what if I tell you that these studies were just not published? So now we have a problem, because actually effect that very likely does not exist in literature is represented as an effect that actually does exist. But publication bias is not the only issue in science. We have different things that can go wrong along the research life cycle. I'll just mention a few. For example, in data collection, if you do experiments, you can have incorrect control group, or you're not blinded to data collection, which is sometimes not possible to blind yourself, but sometimes it is. That means that you don't know where your sample comes from. Then in data analysis, very common thing is p-hacking. I've done it. I'm sure all of you have done it. That means that you analyze, and then you remove some variables, and then you add some other ones, and you remove some data points. But I mean, you justify this to yourself. You think it's justified, but somehow in the end you do get this significant result, and that's p-hacking. So hacking for your significant p-value. And as I already mentioned, often results in published literature are not really well reported, or they're even not reported at all. And the question is why we have these issues, and I believe it's because of a mix between cognitive biases. So as humans, we tend to see patterns where they don't exist, but also we tend to find things that we do want to find. So if you Google something and you type it, does X help with something, and then you find that support for your hypothesis. But this is coupled with a problematic system of science that we work in. And it's based on incentives to publish papers. And you publish papers if you have significant results. And why I put money thing here is, why do we have this system? Because I ask myself, so why don't we change it? And I think it's a lot because a lot of money is involved in publishing process. Publishers, big publishers, have larger profit margins than Google. So they earn a lot of money, and they don't want to change our system. And here, this is a really nice article on some cognitive biases and how to avoid them. I love the Texas sharpshooter. So you ask somebody to blindly shoot at the target. And then you bring in a second person and ask them where this first person has aimed at. And the second person will find a place where more dots are settled, like around a certain spot, and say, here. But actually, there was no pattern. And the second one, asymmetric attention, something that I've done. That means, just like to give you an example. So I did some analysis, and I got a result that I really did not expect. And because of that, I went back to my data, and back then I didn't work reproducibly, so I copy-pasted things in Excel. And basically, I copy-pasted formulas and not values. So I actually got wrong values in my column, in my table for analysis. So I thought, if I did get a result that I expected, I would never go back to my data, and I would never have checked, and they would still have been wrong. So this is asymmetric attention. So we pay more attention to things that we did not expect and then check them. And if we expected them, okay, that's what I expected. I'm not going to check what I've done. And about money, I just want to suggest you to watch this great documentary about Aaron Swartz, who made a lot of, articles from Elsevier and Scopus, I guess. No, Scopus, another big publisher, open illegally, and they actually put a suit against him, and it's like an old story that really shows you how horrible publishing system is, these big publishers. So I try not to publish with big publishers any longer. Okay, so these were the problems, but I'm more interested in solutions. And the first step in finding a solution is to admit that you have a problem and prove that you have a problem. Because I showed you some stuff there, okay, maybe we have some problems, that was a hypothetical experiment, but is that really so? And obviously, if you have solutions, you also need an evidence to test if that solution is really helping. And evidence of a problem sometimes, and it's behavioral research on spiders, where basically Jonathan Proud forged the data that he would give to his collaborators. And that came out because somebody seen the data and seen that a lot of values, so it was published and data were published with the paper, and he has seen that a lot of values are repeating, like exact same values. And he asked the first author, Kate Laskowski, how can this be? And then she went digging into it, and then they discovered what he has done, and I think he later lost his job, and he's not in science anymore. But we can also study, scientifically prove problems. And this is called meta-research, and this is what I currently do. And this is now related to that publication bias thing, so it's not just a thought experiment. These people, and it's about medicine, in medicine they have registries. In registries, you register your plan for your research. A lot of funders ask you this. And then once you're finished, you put results in a registry, and you can also publish them, but you also have to put them in a registry. So these are all results that were published in a registry. Red ones, again, negative results. Green ones are positive. And they looked what was published in journals. And you can see that half of the negative ones were not published, and all of the positive ones were published. And there's something that's called also outcome reporting bias. So in medicine, you can often, we do that too. So you can define your outcome in different ways. So then they would report those outcomes that actually supported their prediction. All the other outcomes were actually not favorable. And in this case, if a doctor would go into published literature, they would maybe recommend, this was for some treatment for depression, a patient to take this pill, because it helps in depression. But actually, the evidence is not very strong. And here I just want to mention, because now I introduce meta-research, this is not same as meta-analysis. In meta-analysis, you find all the studies published on a topic, you extract effects, and you combine them analytically. In meta-research, you kind of also usually work on a large set of literature, and usually you also do meta-analysis partly, but it can be on any aspect of research. For example, we research gender biases in publishing. This brings me to something that we recently studied, and I put a positive term here, unused research, but we called it wasted research originally in ecology. So to sum it up, you can read more in our paper. Almost 45% of ecological research is never published. So we did something similar as them in the medicine, but we don't have registries, so we looked for unpublished studies in different ways. Then, of published studies, so this is peer-reviewed literature, 70% is poorly planned. So they have bad analysis, or they don't have a proper control group. And 40% of results published in studies that are in journals are underreported. So there is a result, and it says, we did not find any significant effect. Still, what was the effect size? What was the sample size? These things are not reported, and they should be. So we calculated that between 82 and 85% of research is wasted. That means research started, was funded, but there is no benefit to the end user. And we based this on more than 10,000 studies in ecology, so it's a large sample size. And I wouldn't trust our results, because they were a bit drastic. If there was no other estimate from medicine, so the only other existing estimate, which is 85% of research, so very similar numbers. And this is health of humans, yes? And it's around $140 billion a year. But there are also things that are more difficult to look at, such as question of research practices. P-hacking is one of them. So people conduct surveys, and this is survey of ecologists and evolutionary biologists. And they ask them, have they ever used one of these practices? So maybe you can't see, but it's like p-hacking, even fabrication here at the end. And this is what they said. So most of them have sometimes done one of these things. But the funny thing, what I find the most interesting, these little dots, this is when they ask them to estimate how often their colleagues do the same thing. So obviously, there should be the same estimates, but they're not. And I believe that then, well, solutions, when we prove, and I think we have a lot of proof we have a problem. I don't think we have to prove it any longer. One of the solutions are open science practices. There are other solutions, but this is what I focus on. And I'm going to talk about 3 practices that I work on or have worked on. One is registration. The other one is open data. And the third one is open analysis, open codes that you use to process and analyze your data. So registration can have 2 forms. One is pre-registration, and the other one is registered reports. Maybe you heard about registered reports. And they both have the same basic idea. So you have your study line, and at the point of study design, you can submit your design to a registry. In medicine, this is commonly what I call core study design. So the registration template asks you, what is your sample size? Did you do power analysis to decide on your sample size? How are you going to blind yourself to data, to your data collection? Are you going to conceal this from patients and similar? And then you go on and you continue with your study, and then you can publish your results in a registry. This is something that does not happen in ecology at this point. Then you have another venue, which is registered reports, where you submit a protocol to a review. So it goes through a formal peer review. Your study design hopefully improves, and then you go on, it's published, and then you go on to do your study. And this is nice, because commonly when you get reviewers' feedback, and okay, if it's about data analysis, you can correct that. But if it's about data collection, I mean, it's really bad. So it's nice to maybe get a peer review before you start collecting your data. And then this goes through stage 2 peer review, and the journal has committed to publish your study no matter of the result that you get. So if it's negative result, doesn't matter. Your idea is good, your design is robust, we are going to publish it. You can learn more from our paper that's published this year. Because we wanted to see, is there evidence that this registration is really making research better? So we wanted to see if these studies that are pre-registered or registered reports have higher methodological quality. Do they have better reporting, more available results, and the features of results are different because you can't study QRPs directly. But under QRPs, these questionable research practices, we expect to find more studies supporting hypotheses, more significant results, and similar. So we collected 26 such studies that compared pre-registered versus non-registered studies that were published, and registered reports versus non-registered reports in these 4 main categories. And most of them were from medicine and psychology, because that's where these things happen. None from ecology, obviously. And to sum it up, yes, all of these studies really strongly support all of our notions. And I just want to show you how, for example, these studies, what they've looked at. So this one is psychology. They compared methodological quality. So there are different aspects. You can't see them, unfortunately, of methodological quality between registered reports and comparable non-registered report study. And this is what they found. So if it's on that side, on the right side, means registered reports are much better. And they obviously are. About these lower QRPs, so these guys, again, in psychology, compared standard reports with registered reports, whether they supported or not their first hypothesis. So if it's light purple, it's not supported. If it's dark purple, it's supported. And I mean, this is really striking difference. So if you don't have registered reports, you always support your hypothesis. If you register report, then in half of the cases, you don't find support for your hypothesis. And obviously, I mean, this can have alternative explanations. Maybe people who do registered reports, they're also more aware of all of these different things. So maybe they just would anyway do their research better. This is something that's much more tricky to test. But I still think there is some pattern is there. And I mean, theoretically, if you think about it, it makes sense that they are somehow different than those that you did not register and that you done whatever you want in your analysis. About open data. I'm sure you're all aware of open data because that's a big thing now, and you have to usually publish your data with your paper. And we were interested in, do people actually use these open data? Have any of you ever used somebody else's open data? Yeah, okay, a few people. And we created a long time ago, in 2018, something that we call resource catalog for finding data. So there we list around 20 places that you can use to find data across repositories. The same as Web of Science searches for articles, these aggregators search through different data repositories, even through supplementary materials. And currently, in our current project, we are developing EcoEvo data detection tool. So you can put a lot of PDFs in of papers and ask this tool to tell you which of them have shared the data, and then automatically download the data. But then, you can maybe access the data, but can you actually use them? And this is a study from Roche et al. In 2015 when they looked into completeness and usability of ecological data. So everything that's 1, 2, and 3 is a low score of usability. So most of the data, even if they're open, you can't use them. And this is our experience too. So case study 1, we wanted to do a meta-analysis on environmental coupling of heritability and selection, because the rate of genetic change depends on heritability and on selection, and environment can change both heritability and selection. And it can act in the same direction, so it increases the rate of genetic change, but also in opposite direction, so it decreases the rate of genetic change. But at that point, there were only 2 studies published on this topic, and they actually had opposite results. So we said, okay, let's find pedigree data that also we can link to some morphological traits, and then we can also maybe answer this based on these open data. So that's what we set to do, and we found a lot of data sets. I don't know why it's so blurred here, because in my screen it's not that horrible, but basically these different colors represent different taxa. Green is birds, obviously, and for example, yellow is insects. So we found a lot of data sets, I think 130. But when we tried to use them, they were horrible. We couldn't use them for different reasons. So we basically ended up with some studies on birds. And we did meta-analysis. We did not find strong evidence that there is environmental coupling, but it's very like a small subset actually in the end of studies. You can read it in this paper there. My case study, too, is something we never published. So I collaborated with some people that wanted to know how meta-neutral communities are structured by their environment and why do they care about these little bacteria or whatever they are. Is because they are absorbing methane from atmosphere, and methane is horrible greenhouse, how you call it, this gas. So these communities are essential because they are only sinks on Earth of methane. And they're changing because of differences in changes in land use. And for this study, they wanted some metagenome data, and there were plenty. It's more than 10,000 data sets we could have used from these main databases of metagenomes. So that was great. But then they also needed some environmental stuff, and they told me this is commonly what you also always measure in your soil sample. And only 20% of studies or these samples actually had these data available. And even worse, for example, this is for nitrogen. Black thing is units were not reported. So you have some concentration, but you don't know what is it. Is it milligrams per kilo or micrograms per liter or whatever? So that we couldn't do this study because in the end we didn't have enough of data. Which brings me to a concept of FAIR data that you might have heard of. I'm not going to tell you what FAIR stands for, but I'm going to show you an example. So if I give you this data set here and I tell you, use it, do whatever you want with it, it's open, you would probably tell me, well, I can't because I don't know, what species is this. I don't know if age is in years or in months, and what is this, FRL? So it's not fair. But if I tell you that this was collected at a certain cave and it's Plecotus auritus, a little bat, on the date and capture method and some other interesting stuff, and if I explain you every single variable, suddenly something that was completely useless to you becomes useful. And this is what's called metadata. So metadata are describing your data set and making it useful for others. And some people have estimated monetary costs of not having fair research data in Europe, and it's huge. It's like 10 billion a year. It's a lot of money. And back in the day when Twitter was not dead, I asked people which one is more important for scientific progress, open data or fair data, and they said it's mostly, well, most of them said it's fair data. But when I asked which is easier to achieve, it's open data because you can just put it in a repository and it's open. But to make it fair, you actually have to explain everything, describe everything, make it machine readable and similar. And how can you do this? And we've done that for Spybird network and database that I have co-founded some years ago. So these are long-term studies of breeding birds where individuals are marked. This is just one example from Hoge Veluwe in the Netherlands. I don't know if this is working. Okay. Anyway, they should show you numbers of broods for these 6 different species. So you can do a lot of things with these data. I'm sure many of you have worked with similar data. They're very important for evolutionary ecology. And more people actually want to combine data from different studies because they want to understand, different geographic patterns. So where is climate change more affecting lay date and similar. But it's very difficult to combine the data because although people collect very similar data, they store it, and trust me, really, I think now we have 150 data sets. Everybody stores them differently, describes their variable differently, calls their variables differently, so it's a mess. So what we do in Spybirds, we take these, we call them primary data from data owners. Then our code developers, they are creating pipelines to convert this into standard format. And this format was agreed in the community. So it's not that we imposed it, we just suggested it, and then people made changes, and then we agreed on a standard format. We create standard format data, and we also describe data with metadata. So each population is nicely described. And then you can go on Spybird's website as a user and request the data. And some data are open, so you can just download them in the standard format. And for some, you need to get approval from the data owner. But people usually approve it, but they ask to be authors, obviously, co-authors. Yes, so that was about fair data. And I thought, like, why wouldn't you as behavioral researchers do something similar? And I'm aware that data sets are inherently different, but there are things that can be standardized. Because an issue when it's not standardized is not that it's not only that it's difficult to combine, but also sometimes it's difficult if different effects you detect in different populations are an outcome of real differences or in differences in the way that you collected the data and stored your data. Okay, and final thing, how am I doing? Good. I always talk quicker when I'm here than when I'm trying at home. So I can be slow now. Open codes. It's about data processing and analysis. And this is something previously that I've done always on my own. I would do my analysis, and then I would discuss my results with my collaborators and blah, blah, but nobody ever checked my analysis. And there is a nice blog from Daniel Bochnik. It's on this link here, where basically somebody discovered after the paper was published that one solitary line of 1 minus that was not supposed to be in a code made their results significant and positive rather than negative and significant. So they retracted the paper. So he asks, why don't we review our code as a part of the peer review process? And a more extreme example is something called most expensive hyphen in history. And that's when NASA tried to launch Mariner 1 to explore Venus. And there was this one single missing hyphen in the code, and the thing exploded. And it was billions of dollars, $80 million, 1.2 billion federal budget for whatever. Anyway, it's a lot of money just because of this line, this one hyphen. And I mean, if you have friends that are coders for companies, they always check each other's codes. They also have some custom software or whatever that checks the quality of the coding, and then they check each other's codes. We don't do that. Now, in my group, we do it, just to make sure that the first person has done it correctly. So yes, my point here is, well, you should do it too, at least within your group, even if you don't make it open. Share it with somebody and made it check. I don't want to check my old codes because I don't want to find out I've done something wrong. Because code is very important for you to understand analysis. For example, as a reviewer, I often ask authors to send me their code because sometimes I don't understand fully what they actually analyzed and how. So they just mention some variables, but I don't know what models they used and how they put these variables in these models. And that helps you to evaluate the conclusions. Also, I reuse other people's codes. I reuse many codes from other people. And I think that saved me like 30 days of work. And I think they increase trust in science. And that example of Kate Laskowski, she said, I was able to discover that I didn't make any mistakes because I worked reproducibly. So she had everything. She had her codes for data processing and data analysis. So she knew that it was not on her end that things went wrong with spider data. So we looked in 2020, do journals in ecology have code sharing policies? And number of journals that do have it increased for these 96 journals that we could compare for, from 14 to 72, which is quite a lot. So they tell you, no, you should consider or you should share a code that you used. But then we looked, and this was really funny. So do people really in these journals share codes? And you can see, so these are two sets of years that we looked at, 2015-16 and 2018-19. Only 20% actually did share code. And these are journals with policy, so people still don't share code. So this is the best case scenario in journals with code sharing policy. Okay, so I told you all of these things, and obviously the question is, why would you care to do things maybe differently than you've done in the past? Because I'm sure, as I did too. I did well without doing these things. I published papers. I think first reason is, in my case, I'm really interested in world around us, especially animals. And as researchers, we are always collecting snapshots. It's snapshots of time and space. And we try to use this to base a lot of research and answer a lot of questions on. And if we impose all of these other issues on it, my question is, how soon, if ever, are we going to get to some knowledge that is actually very important, for example, for solving many of the global challenges? Second, obviously, researchers themselves think that we have some crisis in science. Maybe you heard about these replication projects that largely failed to replicate previous results, started in behavioral research, actually, in psychology, human psychology. And public also sees these things and says, well, I knew that science was wrong, I'm not going to take that vaccine, and obviously they are wrong because when we do things wrong, sometimes they are picked up by newspapers. So we should also think about that. In my experience, because obviously I think research shows that global benefits are always less of an incentive than benefits to ourselves, because we are little selfish people, most of us. So in my experience, what difference it made to me is that I find it much easier to retrace my own research. So if I get requests from reviewers to change something, previously it would take me 1,000,000 years to just understand what I've done 3 months ago. Now it takes me half an hour. So I really save a lot of time. Second, with pre-registration, because I pre-register my research, I now do mostly meta-analysis, so I never pre-register kind of more primary type of work. But it really, all of projects that I pre-registered went so much smoother than those that I didn't, because we thought of everything much more ahead, in much detail, because once you have pre-registration, you don't want to follow it. So we think more about why we chosen certain variables to explain that. Why this one and not the other one? Why are we going to analyze data in this way and not some other way, and similar. So everything went much easier after that. And finally, I really like the community of open science people, because it's very kind of welcoming, and I met a lot of interesting collaborators that way. And obviously, I also suggest you to watch this plenary talk. Is online. It was Society for Open, Reliable, and Transparent Ecology and Evolutionary Biology 2024 conference by Kate Laskowski, who was that person involved in Proudgate, because she there describes how they came to realize what happened with the data. And I think that open science practices, if data were not open, they would never have realized this. So that research would still be there and not retracted. And it was wrong. And obviously, we also need a different system. We need different incentives. And this is a pyramid of culture change from Brian Oshek from Center for Open Science, where if you want people to do something, you first have to make it possible. So you have to make infrastructure. For example, if you don't have data repositories, you don't have anywhere to put your data. But also very important are incentives. And we don't still have incentives to do reproducible research. And that's a fact. I do it because I like it, and I think it's better. But yeah, I understand if somebody doesn't want to spend time on making their data fair or making their codes open. And I think, did I want? No. Okay, and what can you do to make things different? I know what I skipped, sorry, because I put it on a different slide. Here, I want to put you a YouTube video also on why. This is about making publications open from that little documentary that I mentioned. It's going to play.
I wish we could change the past, but we cannot.
Oh.
But we can
Oh. It's okay.
It's okay, just carry on. You had a hyperlink in that slide somewhere, but it's okay, just carry on.
Okay, with the video or just with the presentation?
Just with the presentation.
Okay, pity. Because it's a nice video to show you why, also to make your research public. And it's about, so there was this kid who read a lot of publications because this guy, Aaron, made them open illegally. He was a high school kid. And based on this, he had this idea of developing a new way for early detection of pancreatic cancer. And then he did a little research project on it, and then he contacted some medical whatever developers, and they liked it. And they actually did develop a proper test for pancreatic cancer based on something that a high school kid has discovered based on open literature. And I always think of it, you never know who can use your research, be it published research or maybe your research data, to come up with something that's actually very revolutionary or something very important for, in this case, saving somebody's life. So for me, that's also another reason why I think we should strive to be more open. And what can you do? So with registration, so I just list some ideas. Obviously, it's not easy to start with new things. There is a lot of workshops now. I give workshops on registrations for free online. So if you want, you can always invite me to give it to your group. It's short, it's half an hour to show you how it actually works in practice. You can obviously read about it. There is more and more publications on registration and how to do it. Try it. I think this is one really good way to learn and to see if it's going to work with you. Try it with your student. I do it. Now my PhD students, they have to register everything, so they don't like it. But they will. I also didn't like it when I started. You can also become a recommender I'm a recommender for this peer community in registered reports. Recommender is something similar to reviewer, because it helps me to also understand the process, for example, behind registered reports and how it actually works. What can you do with data? Again, very simple. Describe all of your variables. So they are easy, and give it to your colleague and say, hey, I give you this data set. Do you actually understand it? Try to use some general standards if they exist. For example, ecological metadata language, but you also don't have to. Just describe it. Find a repository to put your data in. This can be a discipline-specific, for example, Global Biodiversity Information Facility or Flybase, or it can be generic, like Zenodo or Figshare or Dryad. And this, I don't know, re3data.org, they list all the repositories by subject. And I checked, there is none for behavioral ecology or behavioral animal behavior and similar. So maybe you can try to start with such a repository. And in the end, you have to choose your license. So terms of use of your data. So data can be open, but you can still put some terms of use. And a commonly used one are these Creative Commons licenses. So for example, you can prevent or say, I don't want this data to be commercially used. So you can't use them to, I don't know, earn money. And you also have today journals that publish data sets. For example, scientific data and gigascience, and I've chosen here two that are related to behavioral ecology. So first one is high-resolution ethnograms of these like Japanese quail, and the second one is a data set of ant colonies to study clustering behavior. And these journals have like high impact factors, around 10. So basically publish your data. The article itself just describes the data and how can you use the data for what types of studies, and then you also have to put obviously data in a repository. So what to do with the code? Again, just describe somewhere, either in a separate README file or at the beginning of the code, what your code does. And what I like is to put some, well, now I use R Markdown, but if you don't, you can just put some inline comments and says, testing hypothesis 1. So then when you go back to your long code, you know which part you need to change or work on. Also very important to write down the version of R that you have, or any other software that you have used, and also some basic information on your computational environment. And you can do this by just typing session info after you run your session, and then copy-pasting that into this description of what your code does. So this is like really basic. This is not, this is what I would tell to somebody who doesn't want to invest time into making their code reproducible at a high level. And again, give it to somebody to run. So we have done reproducibility hackathons where we have given code and data sets from ecological papers to people to try to rerun. And most of them do not run. So they like break somewhere. And when they do, in some cases, they don't get the same result. So for me, this is really like, why? Because you run the same code on the same data. Why don't I get exactly the same result? But they get different results, not always, but sometimes. So just try to give it to somebody to run, to see if it runs and if they get the same results. And again, put it in repository. Maybe somebody here uses GitHub, but it's not considered as permanent place. So what you can do is link your GitHub with, for example, Zenodo or Dryad, and then it gets DOI, it can be cited, and it's kind of permanently stored. And you can also give a license to your code, and I don't use it, actually, but there's this Apache type of licenses that is recommended. And Ed, a colleague, he has run this collaboration to help people to implement code review in the scientific workflow. So if you would like to do it within your group, I think this is a very simple guideline on how to do it. There are also reproducibility teas. They're everywhere now. You can also join them online and participate online. And it's basically kind of like journal club for open science. So people read papers, and then you discuss them in similar. There are also you can read meta-research articles on all of these different topics. And many journals now, like eLife, PLOS Biology, they have a section that's called meta-research. Also, I like this Metrics Forum. It's a Stanford Meta-Research Center. They have great talks. You can just go on their website and subscribe. They're often not in like ecology. They're from different sciences. Sometimes it's ecology. But what I like here, I get a lot of cool ideas from them. And maybe you also want to do some meta-research in animal behavior. Because how I see the role of meta-research is that, so this circle is what we could know based on all the research that's being currently done. It's a lot. But then what we learn in reality, because all of these inefficiencies, unpublished stuff, is kind of small and a bit out of the real truth. So meta-research wants to put this back into like true effects and also increase the knowledge, potential knowledge that we gain from currently done work. So not some extra things that we can do. And finally, maybe you also want to join some societies. I mentioned two that might be of interest to you. One is SORTI, Society for Reliable and Transparent Ecology and Evolutionary Biology. The other one is interdisciplinary meta-research something. They're both really nice, and people are very friendly. And finally, if you do things in this way more openly, I learned that I do it my way because I have opportunity to do it. And this is what I've chosen. It worked for me. And I'll try to help others, but definitely don't judge others because they don't do it that way. And at the beginning, I thought, why not everybody would do it this way? But then talking to people, I realized it's not as easy for everyone. And just an example from SpyBirds. So at the beginning, I wanted all the data to be open in SpyBirds. And big groups were pro it. So they said, OK, you can make our data open. But small groups didn't want that. Why? Because they're small. Their funding usually depends on this data they're collecting. They can't publish a lot of paper in a short time because it's a few of them. So they don't want opening their data because somebody might do this before they manage to. And big groups, they don't care because they anyway have like 20 PhD students and they're working like machines. So then I realized, well, really it's not the same for transparent and open way. And to end with Charles Darwin, who was mentioned in somehow negative connotation today, what he says here, that I had also during many years followed the golden rule, namely that whenever a published fact, a new observation or thought came across me, which was opposed to my general results, to make a memorandum of it without fail and at once. For I had found by experience that such facts and thoughts were far more apt to escape from the memory than favorable ones. So basically, it's like we remember things that just fit into our worldview and our world frame. And with that, I would like to thank to Spybirds, obviously, to all of these my collaborators that made things that I've showed possible, and also to you for coming and listening and for inviting me to give this talk. And of course, I'm open for any questions. Also want to mention, if somebody would like to do some meta-research here, you can always contact me and ask me for some advice or whatever. I would really love to help. Or if you want to set up animal behavior repository.
Okay, well, when we put forward the abstract for this, we didn't know that it would actually be published by now, and as you can see, it was conveniently published very recently in Science. Well, it's now a quarter of a century since I and some other authors published a paper which was the first systematic review or survey of chimpanzee cultures in Africa. And what we reported there was 39 putative cultural variations right across Africa, traditions, if you prefer. However, that's increased over recent years. More of those have been reported, and certainly many more field sites, which is partly what I'm going to talk about. But there hasn't been any change in those putative cultural behaviors. And you can contrast that with the position in humans, where the kind of cultures that we express build up generation by generation what went before, and in general they often get more complicated and more efficient or more effective. And you can think of just countless examples of this cumulative culture, sometimes called cumulative cultural evolution. Here's a couple more examples. Certainly in technology we can think of that, whereas chimpanzees have been studied for about 60 years now, and we haven't really seen any changes, certainly not changes at all like these, in chimpanzee behavior. So some authors, in fact I would say it's kind of received wisdom now, that it's cumulative culture that distinguishes human culture from all the rest. Cumulative culture is unique to humans, it's been said. However, people who study chimpanzees in the wild see behaviors like this I'll describe and think it's difficult to imagine that it couldn't have been cumulatively or couldn't have been produced without some cumulative build-up. So here we have a chimpanzee in the Congo region of Africa, and with a small party that perhaps approached some clearing, they're already carrying stems in their mouth, often much longer than this, maybe a meter long, several of them. They know what they're going to come and do, but the first thing they do when they get there is pick up a stout stick, like illustrated here, and they push it directly down into the ground, which is often very hard, and it takes them many minutes to really push it down using 3 limbs to force it down like a spade. They make a tunnel down, then they remove that, and then they take one of these stems, they fashion it into a brush tip at the end by stripping it through their teeth, they wet it, they form it into a point very carefully and feed it down that tunnel, and then pull it out with some termites biting on it, which they nibble off. So what we have here is what we're calling a complex tool set, using one tool and then another tool in this case. And it's difficult to imagine that that just came out of the blue rather than built up cumulatively, particularly culturally cumulative, given all we know about chimpanzee social learning and culture. The problem is, of course, that these materials are perishable. So we don't have any kind of historical record the way we do for, let's say, the wheel in the case of human culture. So in this paper, we've tried to take slides are not advancing. Okay. In this paper, we try to take a different approach by using contemporary discoveries in genetics, particularly about the genetic similarities and differences amongst chimpanzees across Africa, as a kind of genetic time machine to sort of understand what happened in the past. And we're now able to use 35 different locations where we have both information on the genetic similarities and dissimilarities between chimpanzee populations, and we also have behavior on their whole behavioral repertoire. And this comes from something called the PAN-AF database, which has been built up by many researchers over many years, initiated by the late lamented Christoph Busch. So from that, what we can do is produce the pictures of the genetic links. Here's one of the genetic markers used, and here's another one. These range, they're quite recent, over 5 to 15,000 years. And the reason we're seeing those is that in chimpanzees, as in many species, one sex or the other moves. In the case of chimpanzees, it's females who, when they reach sexual maturity, tend to move to a different community. They avoid inbreeding. And of course, they take their genes with them. And then over centuries and millennia, the genes spread, sometimes far and wide, as you can see from those diagrams there. But those chimpanzees, those female chimpanzees, may also be culture bearers. And so as the genes spread, maybe you'd predict that the cultures would spread. And that's what we looked at in this paper. So we divided the behaviors into 3 main categories. There were simple behaviors that didn't involve any tools, which might be as simple as just plucking a berry and eating it. Then there was what we call simple or single tools, which are things like this masticated ball of vegetation that's used to pick up water from a tree hole and drink it. Or there's these complex tool set behaviors, like the one I described about subterranean termite fishing, or another illustration you see there is using a stone as a hammer to crack nuts on a stone anvil. And you can see from the picture at the bottom the idea of what we're looking at here, using the genetics to predict, well, where females may have moved in the past, and did they transmit these cultures in these 3 categories. And what we find is that if we regress those behaviors on the genetic connectivity, population connectivity, it raised the odds of sharing the complex tool sets by a factor of between about 2.5 and 5.5. And that's what you see illustrated in the part of the graph at the bottom there. In the middle is those simple single tools, and here the effect was less, something like a factor of 1.5. And for the non-tool behaviors, there was no such effect. So our inference from this is that the complex tool sets are invented relatively rarely, and their adoption and spread is highly dependent on transmission by those knowledgeable females who are moving from community to community. The second of 3 analyses then builds on that. It's called something called reinforcement analysis, which was new on me, a statistical technique, which to some extent, you might argue, goes beyond, well, you can't tell causality in a correlation to address something about causal directionality. I'm just going to give you our conclusions from this. That was when the complex tool sets are shared between populations, we find it's then highly probable that the genetic markers are also shared. And yet, interestingly, the converse isn't true. Sharing genetic markers does not guarantee, or it doesn't even really significantly predict, sharing the tool sets. There can be genetic transfer, but the culture is not necessarily transmitted. So our inference from that is that spread of the complex tool sets is dependent on cultural transmission, as indicated by the genetic markers, but the genetic links are not sufficient to predict the behavioral similarities. In short, those behavioral similarities don't depend on the genetic differences themselves. There's then a third analysis. Oh, okay, just to add in, of course, in the paper there's lots of stats and graphs and so on. Spelling this out, I don't have time for that. If you look at the 2 top left graphs, that's where you see the effect I described, and in the 2 at top right, you see where, when we look at it the other way round, we don't get the effect. The third analysis showed that the complex tool sets are linked to simpler versions in other populations. So I described that fishing for subterranean termites using a tool set. Then we find there are other places where they're only using a single, more primitive tool to do some digging and get at subterranean food sources. And we find that they're all linked to the more complex ones along these genetic pathways, if I can put it that way. So here are some examples of that. And again, I don't have time to go into that here, any of those details. But it just so happens we also have a poster relating to this study that you can look at later, and that has more information about that. I can tell you more about it. So leaping on to our conclusions, and this gallop through the study, That first analysis, the regression analysis, showed that the complex tool sets seem to be invented relatively rarely, and their adoption and spread's dependent on transmission by the knowledgeable females. That was our conclusion. And then the reinforcement analysis reinforced that. Spread of the complex tool sets, we concluded, is dependent on the cultural transmission as indicated by the genetic markers, whereas genetic links are not sufficient to predict behavioral similarities. And then the analysis that I just talked about shows that the complex tool sets are always linked to populations elsewhere that show those simpler versions, suggesting that the complex version arose by building on the simpler ones. Or repurposing, that's to say, shifting from using the tools for one purpose to another purpose, like those two examples there. And you don't have to take our word for everything here. There is a nice perspective piece in the same issue of Science, and I've got a copy of that here for those who'd like to read it when you come and look at the poster, by Pete Richardson and Rob Boyd, who, as you may know, are two of the founders of the whole field of cultural evolution. So it's quite an honor to have their evaluation and context setting. And finally, I just want to finish by thanking those responsible for these very large now genetics database and behavioral databases. They're based on a huge amount of work by a huge number of people, such as for the genetics one there, the 88 co-authors in a paper by Claudia Fonseca et al., and for the behavioral database by Ami Callan and colleagues, 68 others there. So we thank them for that. And actually that touches on the question that Roman Wittig just asked our previous speaker about what's the proper way to go about this. So we're using those public databases. Thanks to those who put those out there. That's me done.
Okay, well, today I'm going to talk about how we use visual attention to draw inferences about cognition. And basically what I want to do is talk about the ways in which we can try to improve how we go about this. So a lot of work that helps us to reveal insights into cognition actually comes from studies on gaze. So essentially what we're doing is we're measuring visual attention, and then we're trying to use it as a proxy measure for cognition to try to understand how our study subjects respond to a particular stimulus, basically in their social or their physical environment. And through studies of attention, we've actually really been able to get to ask some questions that otherwise we maybe wouldn't. So we've been able to ask about perception of causal relationships, mental state attribution, memory, so quite big questions. But more and more what we see actually is that people are kind of going down the eye tracking route to do this. So this makes sense because eye tracking is a really great tool that allows us to kind of increase the precision in our measurements and to even track attention over time. But the problem with eye tracking is that it's very expensive. It doesn't necessarily work for all species, and it certainly isn't implementable in every setup, especially if you're working in the field. So today I really want to focus on the use of the measure of visual attention in the absence of eye tracking. And when we're going about this, what are the sort of considerations we need to make to improve the way that we do this? So these are sort of the 6 points that I would like to cover. For the sake of time, I'm actually going to focus just on 3 of them, but I do have more slides, so if you've got questions at the end, we can always talk about these other points or in the break, or I can refer you also to the papers at the bottom of the slide. And so of these 3 points, I want to start by talking about paradigm choice, because I think it's quite key to highlighting some of the issues that we get when we're measuring visual attention. And there are obviously different paradigms. I've kind of put some of the main ones up here, but the one I want to focus on is the preferential looking paradigm. So the preferential looking paradigm, it's a common choice in cognition studies because it's easy to implement and it's easy to measure the response. So essentially what it involves is presenting pairs of images that differ in some particular variables. So the example here is 2 different species of macaques. So for example, we're measuring ability to discriminate between species. And these images are either presented together at the same time or individually. And then we simply measure how long our subjects look towards each image. And so if we see that there is a sort of a bias in visual attention between these image pairs, we can interpret this as ability to discriminate between these stimuli. So that's great, but usually we want to put this in some kind of cognitive framework or social behavioral framework, and then we start to sort of make broader inferences. So one thing we find is that if we find that our subjects are looking more at, let's say, the in-species group, we say, oh, okay, well, perhaps they have a preference for familiar facial features because they have socially relevant cues. So that's why they're looking at those pictures for longer. But what if they're looking more at the out-species group? Well, then we can say, okay, maybe they have an interest for novel facial features, and that's why they're looking more at the out-species group. So we have to be really careful here about how we structure our hypotheses to guide our data interpretation and how we make those interpretations. And I think it's just really important to emphasize that the inferences that we make about cognition are really based in, really depend upon the paradigm that we use and the measures that we take and the hypotheses that we build around these. And so there are, of course, some limitations. So one thing is that when we see a visual bias between stimuli, there is a tendency to infer preference, often social preference if we're looking at social stimuli, but these two are not necessarily equated. So the presence of a visual bias doesn't necessarily explain why there is a visual bias. Another issue we have is we often don't think about gaze avoidance. So if you present two different stimuli, perhaps our subjects are looking at one more than the other because one is aversive in some way, and so they want to avoid it, so they end up looking more at the other one. And then another issue is that actually the absence of a visual bias does not necessarily equate to the absence of discrimination. So this is especially the case if we're taking global looking time measures. We're not thinking about the particular sorts of individual features of a stimulus that might be drawing their attention. So we do need to be careful when we're addressing cognitive interpretations in visual attention studies. But the good news is there are steps that we can take to try to improve how we do this, and this is what I want to talk about now. So one thing we can do is consider our measures. So we don't just look at overall gaze time, for example. So Aslin refers to what he calls the microstructure. So for example, measuring other aspects of gaze, such as how often or how long our subjects look away. Again, considering avoidance behavior. So if there's something aversive or threatening in the stimulus, taking this into account is important. And also thinking about how our experimental data map to real-world data. So kind of giving them a grounding in ecological validity and trying to understand why a stimulus would elicit a visual bias. And so I think there are a couple of nice examples to highlight this. So in this study, it was done with infant rhesus macaques, and the authors basically showed different types of video stimuli to these infants, and they found that the time they spent gazing, particularly at the eye regions of this example on the left, was correlated with the time they spent interacting with their peers in their social group. Another nice example comes from work that's also been done in rhesus macaques on this male facial skin coloration. So this has been proposed to be a sexually selected trait. So the idea is that males with redder faces have a higher fitness and are more attractive to females. And so initially this was tested in a sort of an experimental setting where the authors measured how long female rhesus macaques look to images of different facial redness, and they found that they look longer to the redder faces. And what's nice about this is this was later kind of corroborated by observational studies in free-ranging macaques. So actually, they found that females were more likely to solicit matings with males with darker, redder faces. So this, I think, is a nice example of how our experimental studies can complement our observational studies. Another thing that we can do is to validate our measures within experimental context. So in this example, again, it's with rhesus macaques. The authors measured gaze duration to images of conspecific faces with different facial expressions, and they showed high repeatability across a 4-year period, even despite the fact that they used different testing setups. We can also think about how different experimental measures correlate, so within that sort of that testing paradigm. This is actually an example from a study I did with long-tailed macaques. So we were interested to try to measure whether their looking time in one study was correlated with their choice of images in a separate task. So basically we measured gaze in one and then choice in a forced choice task separately. And actually, for social images, we found there was no correlation at all. They didn't show strong preferences for social images. But when we looked at non-social stimuli, we did seem to find a quite consistent pattern. So they were more likely to look at images of food and objects over landscape, and they were also more likely to choose images of food and object over landscape in the forced choice task. So we saw this nice consistent pattern here. And then the final example I would like to come to is also about validation. So again, this is a study that used sort of different experimental techniques. And so they did this with horses, and they were interested in how they responded to different facial expressions. So on the left, you have the sort of positive expression with the ears forward, and on the right, you have the negative one with the ears back. And they found that horses were much more likely to approach the images where they had this positive facial expression, and they really avoided the negative images. And actually, in a separate task, they measured their gaze to these different images, and they found they had no gaze bias at all. So this comes back to what I said earlier about the discrimination of stimuli, because in fact, if they had only relied on their gaze measures, they could have come away with the interpretation that these horses don't differentiate between these different affective states, when clearly they actually do. So I think it's really important to think about these different aspects when we're designing our studies, when we're collecting our data, to try to improve the way that we can make inferences about cognition and sort of understand this more broadly. So our paradigm choice in particular and the measures that we take, I think, are really, really important for this. So I think I'm still within time. And I would just like to say thanks very much to ASAB for supporting me with the childcare grant. And otherwise, I think we have maybe some short time for questions if anyone has any questions. Thanks.
I'll run through a series of studies with some postdocs and PhD students, Kate, Laura, and Andy, that I've been fortunate enough to work with, to really explore the impacts of habituation on behavioral data collection. So back in 1948, Kinji Himanishi began a study of Japanese macaques that established a set of methods, habituation, individual recognition, long-term data collection that have become the foundation for behavioral research. Within primates, perhaps popularized by Jane Goodall in her pioneering work on the Gombe chimpanzees, habituation has essentially formed the foundation for much of the primatological research that has happened over recent decades, including some of the longest-running behavioral studies, such as that established by Jean and Stuart Altman in 1963 in the Amboseli Baboon Project. But habituation is not just the foundation of primate behavioral research, even though it's almost ubiquitous in terms of a method in primatology. But a number of long-term studies of a variety of birds and mammal species base their behavioral data collection on habituation. But rarely do we come to consider or think about the biases or the considerations that we need to take into account with these sorts of data, despite the fact that the Strange framework highlights habituation as one of the factors that we need to be aware of. Now, in this context, studies consider, and I've just chosen one chimpanzee example here, the idea of habituation is that our research animals come to accept us as researchers. They ignore us within the environment so that we're able to collect data from close quarters. But this essentially embeds two assumptions. The first of which is that we become a neutral part of the environment in which we're working. And the second is that habituation is relatively equal across all of our group or study subjects. But rarely are these tested. So I want to draw on a couple of, or three examples of studies that we've conducted within a study system I've been working in now for a number of years in the Soutpansberg Mountains in South Africa, drawing on the arboreal Simango monkey initially before moving on to a couple of studies of the terrestrial Chacma baboon. And the things to be aware of within this study system is that these two primate species are exposed to a full predator guild and that their interactions with humans are relatively benign, largely interactions with our research community. So the first time that we became aware that habituation or the process of our habituation and behavioral data collection might influence the data that we're collecting was when conducting a giving up density study to titrate the landscape of fear or the perceptions of risk in Simango monkeys. So these studies are based on a depletable artificial food patch, and the idea is that when an animal feels at risk, gives up earlier, it leaves more food behind in the patch than when it feels safe. It feels happier to deplete the food resource. So for the arboreal Samango monkey, we predicted that they would feel at risk on the ground, where they're exposed to terrestrial predators, much safer higher in the canopy. And that's indeed what we found. So the giving up densities were much lower on the ground than they were higher in the canopy. The challenge was that this result changed fundamentally on the days that we were studying the Samango monkeys and collecting behavioral data. Under those conditions, the animals felt safer. Their risk perceptions, their giving up densities declined to the extent in one of the two study groups that vertical landscape of fear disappeared entirely. Predators were present during that study. They were visiting our experimental setup during the night. Effectively, the Samango monkeys were perceiving us as some form of human shield or a cue to them, the situation being much safer when we were present as researchers. And this also seems to extend to their responses to predator interactions. So we collected fecal samples to measure the physiological stress responses following days with the monkeys, where we recorded the number of alarm calls, the group-wide alarm calls, indicating a predator interaction. And in this particular condition here, you can see that on those days when just a single observer was present, as the number of alarm calls increases, so does that cortisol response. But as the number of researchers increases that are studying the monkeys, that stress response disappears to the extent that when 3 or 4 researchers are present with the monkeys, the following day, when we collect that fecal sample, there's no evidence of the number of alarm calls influencing their physiological response. So in this context of predator-prey interactions, we clearly are not neutral within the environment. But what about more subtle impacts on the behavior of the study animals themselves? Well, here I turn to some great work by Andy Allen, who formally tried to test these 2 assumptions of habituation. And he did that through conducting a series of flight initiation distance approaches, often used as a proxy or as an experimental stimulus of predation risk. So the idea here is that as a researcher, you walk towards your study animal, and the 2 measures that I'm going to focus on here are the visual orientation distance, the point at which that animal looks towards you, and then the flight initiation distance, the point at which it displaces. So the first thing we can ask is actually, how are humans within this social group treated? I won't go into how we generated the predictions of the types of responses that we would expect if they were equivalent to a predator or a social threat, but just to highlight that in the vast majority, over 98% of the approaches that we did, the response of the animals was to treat us as equivalent to a high-level social threat, a dominant animal within the group. Again, we are not neutral. Now, a number of variables help us determine what those visual orientation and flight initiation distances are, such as the behavior at the start of the approach, the habitat type, the number of neighbors. But statistically, the far most significant factor determining an individual is individual identity in determining the visual orientation or the flight initiation distances. Those measures were distinct between and consistent within individuals. Furthermore, the flight initiation distance and visual orientation distance were highly correlated with one another, which alongside other additional analysis is highly indicative of some form of tolerance trait. And that's even if we look at these measured in independent data sets. So what we have here is a set of highly intolerant individuals, mostly within this group are adult females, but as you can see across this tolerance range, age, sex class doesn't help us understand the variation in tolerance. We have a range of highly tolerant individuals comprising adolescents, adult females, and juveniles. And this can have profound implications for the data that we collect. If some individuals, such as this individual, are wary and others seem to be not particularly concerned by the presence of an observer. So to finish with, I want to illustrate how this might impact some of the measured variables that we collect, particularly focusing on social networks. So the y-axis on each of these graphs is the frequency of occurrence as an individual, as a neighbor of a focal individual within a focal sample. And the x-axis here is the tolerance of the individuals. The more tolerant individuals are to the right. I'll focus here on the lower panel. The top panel just indicates that these relationships are consistent across 2 years of study. So first, let's take the situation if we collect our data from over 5 meters or over 4.5 meters away from the study animals. These are the blue lines, and you can see that tolerance doesn't influence here at 5 meters within a 5 meter buffer, or whether these two individuals are touching, they're grooming one another. Tolerance, across that tolerance gradient, there's no difference in the probability of an individual occurring as the neighbor of our focal animal. But if we collect our data close to our study animals, what we find is that intolerant individuals are underrepresented, and this is particularly the case within these grooming dyads. We're effectively blocking those intolerant individuals coming towards our study animals and biasing the data that we collect. And this is represented, and you're going to have to trust me because it doesn't look like the grey lines that have come out. But on my screen, we have a very dense social network here on the right and quite a distributed social network here on the left. Come and see me afterwards, I can show you the original data. But it massively influences the data that we collect and the measured social networks that we might derive. And these, of course, fundamental for a large number of different types of analyses. So even when animals are fully habituated, and these baboons have been studied for over 10 years and considered highly tolerant of humans, clearly research presence has huge impacts on primate behavior. But this goes beyond just the behavioral changes that we observe in our study animals, because we're also influencing their ecological communities, their interactions with predators, interactions with other species, even competition between social groups, depending on the nature of the habituation. And yet the biases that I've reported here are largely ignored. They're largely overlooked. We don't consider the impact that they have on the published literature. So with that, I would like to say thank you to all, and happy, if I have any time left, to take a question.
Yes, there is some time. Be patient.
Thank you. So it's going to get a little meta now as we talk about animal behavior conferences at an animal behavior conference. But we'll be talking about, well, I'll be talking about our experience of moving ASAB conferences online and what happened in terms of increasing the accessibility of these conferences as a result of that. As you can imagine, there were a lot of people involved in this. We did a lot of conferences. A lot of people helped organize these. So many thanks to those. I've already realized I've missed quite a few names off this. But this was funded by ASAB, and it was funded by ABS for some of these conferences as well. And with me with these talks, a huge thanks to Joe Madden, who, as ASAB meeting secretary, helped all of this. And then Jess Cusick at Utah, who helped on the ABS side, and Kirsty Graham, who helped on the ASAB side with all of these conferences. So we've heard already there's kind of a drive and bias within the research community within animal behavior. And this can have an influence on the biases that happen with the research that occurs with animal behavior. Diversity in perspectives can really help drive new ideas and new perspectives on how we do our research. And so we can see there is a bias in terms of the geographical spread of where research comes from in animal behavior, and then drivers within changes in how we perceive and do research. And conferences play a role in this. There's a reason we've all come to this conference today, and that's because we want to share our research. We want to do some networking, and we want to maybe establish some training or develop our skills. And when we have conferences that are not accessible to everyone, it helps perpetuate these biases of not allowing people access to these, or not allowing people to share their research and have a stake in that conversation. So we had a bit of an accidental experiment take place. I don't know if you remember what happened in 2020.
But it changed the shape of conferences just a little bit. First, I'll point out in 2018 and 2019, we were really lucky that for some reason, Joe decided to collect some data on delegates that were attending in-person conferences. We had no idea that this was going to be useful when it came to future conferences because the following year, we ended up having to move these online. My fieldwork got cancelled because of COVID, and so I stepped on board to say, yeah, sure, I'll develop a website for conferences. I didn't know how I was going to do it, but away I got started in May, and then in July we hosted the first ASAB online conference. We continued this, and for the December we had a virtual conference in the December. And then 2021, all of those conferences moved online as well. And we also hosted the first Twitter conference. And I'll go into details about these in a bit. From the first online conference, we published papers of our findings to help kind of talk about this accessibility. So I'll talk about those. And then in 2022, everything started to move back in person, or at least in hybrid. In 2023, we hosted the second Twitter conference. It was part of the start of the death of Twitter, so variable results. But we also published the findings from that too, which is also, yes, people are moving. So for those of you that didn't attend these virtual conferences, I'll just give you a very brief overview of what these looked like. So firstly, we tried to replicate much of the design that we see with in-person conferences. So we had talks, we had workshops, we had Q&A sessions, and we hosted these via Zoom. This is me sat as close to my internet as possible to try and make sure that everything would stream okay. And then we also tried to have some kind of interactive elements. You'll see a variation of this poster room at this conference with the virtual posters that have been hosted. And we organized some of the social events as well. And in some of the later designs, we managed to bring in a bit of a virtual reality aspect. So some of you might have been avatars and met that way rather than in person, just to add a bit of dynamic there. Twitter conferences are slightly different. So rather than having a verbal presentation, you can have videos in the form of tweets and list those tweets. They stay online for as long as Twitter stays around. And so you can still answer and ask questions on there now. So we moved these conferences online, but what did we find happened? So first, there was a huge increase in participation. We went from having about 200 and 170 delegates coming to the in-person events. That increased to about 400, 480 delegates attending the online conferences. Many more probably would have attended too. They visited the website on the day, but didn't have pre-registration. And the Twitter conferences, just absolutely mad. We had nearly 2,000 website visitors. We estimate with hashtag analysis that we had millions and millions of views of these tweets. We also saw that there was an increase in the global reach of the research that was presented. These are the delegates for the in-person conference, with 25% being international. That increased to 55% being international for the virtual conference. This is a Twitter conference here, and this is kind of the histogram of abstracts placed across a map, so you have an idea of the geographical range of the presenters. There's still a little bit of a geographical bias, but we certainly see a kind of geographical spread. These were 24-hour conferences, so we really hoped to make them global. And we also saw that a lot of this was due to the fact that these were a lot more accessible. And we asked people, do online conferences provide you with more inclusive access? And 70% said yes, yes they do. Many people felt like this because they felt like they could afford to attend the conferences. There will be people unable to attend this conference today because they can't have the cost of travel or they don't have the time to travel as well. And we saw that when we asked them about this increase in accessibility, we asked them what made these more accessible to them, and the cost and the time was certainly some of those. There were some variable results as well, so some people strongly agreed that online conferences were better if they had caring responsibilities, but for others, it made it harder. So if you are having to care at home, whereas you could travel for a conference, you're having quite different experiences. So, is it that simple? I've solved accessibility in our conferences. fantastic. No, unfortunately not. There is a reason that we've come to these conferences today, and I imagine a lot of us will get a lot of benefit from meeting in person and talking. Socializing and networking were consistently viewed as something that wasn't quite the same in a virtual space. Even with the virtual reality avatars, just doesn't quite have the same impact. And of course, some people either don't want to spend time on the technology or are not comfortable using it and don't see any value in the virtual conferences and would not attend them anyway. But there is still interest in making sure that there are virtual elements to conferences. This is the geographic range of participants in the first Twitter conference, where everything was still online, compared to when everything was back in person. And we see that although the numbers in those participating in North America and the UK reduced, there was still a huge geographical element in people wanting to participate in these virtual conferences and online conferences. And we see that today with this conference. We have people joining us virtually and the hublets that are part of this conference too, so special shout out to them. So yeah, when we moved these conferences online, we saw an increase in the number of delegates, increase in the global reach, and the increase in the accessibility. So happy to take any questions. Thank you.
Thank you, Amanda. My name is Friederike Hillermann, and I am representing Spybirds today. You've heard about Spybirds already by Anticia's plenary. So Spybirds is a database and a network, and it's connecting researchers involved in studies of populations of individual birds. And I want to showcase Spybirds as a valuable resource for helping you find data and combine data and analyze data from different study sites and study species. And this presentation might potentially even encourage one of you to found your own spy initiative. But let's start by talking and asking why such initiatives that bring together data in a standardized format are important. So imagine Shadow Grove Woods. Shadow Grove Woods is where ecologists have been monitoring bat populations for decades, and in recent years they have been noticing a troubling decline in population numbers. So obviously we want to know what's going on there, what's causing it, the populations to decline, and we want to come up with appropriate conservation measures. So the conservation efforts will be informed by our research, by our conclusions that we draw from our research. And obviously the efforts are going to be different depending on whether this is a local or a more wide-scale issue. So is it a local issue such as reduced insect numbers due to use of pesticides or the pond drying out? Or is it something due to climate change, so that climate change is disrupting the hibernation and the reproductive cycles of the bats? Or is it a disease? Is it spreading? So if we only focus on Shadow Grove Woods, we might miss some opportunities on catching potentially broader trends. And we might also miss the opportunity to learn from other sites. And our conservation strategies might not be the most efficient. So to direct our conservation strategies, we want to know whether other populations are affected too, or whether this observation is site-specific. So achieving this requires some sort of data sharing and collaboration amongst researchers. And animal behavior researchers have increasingly recognized that the power of data sharing, and there is a growing awareness for that our study, the choice of our study subjects limits the generalizability of our findings. And the STRANGE framework encourages animal behavior researchers to really ask themselves repeatedly, just how strange are my study species? And yeah, this should be disclosed in the methods, and it should be considered in both the study design and the discussion of the results. And then there are the many individual initiatives. These multi-lab collaborations, mainly in comparative psychology, increase the number of individual studies, and they diversify the population studied to a degree. So these efforts are important, and they're improving the generalizability of findings to a degree, but there are other aspects to consider too. So before I move on to the next steps towards generalizability, let's clarify some of these important, distinct key terms related to generalizability. So first off, we have validity. Validity is fundamental to good science. Validity means that the study design and the data analysis are accurately measuring what we intend to measure, and that the results are interpreted accurately. So in other words, if the experimental design is flawed, or if the results are being p-hacked, then the results cannot be trusted, let alone be applied more broadly. Then next step is reliability, and reliability refers to the consistency among observations. So reliable results are repeatable, but they may still only be specific to a particular system, like a particular context. And then beyond reliability is generalizability that ensures diverse environments. Okay, now moving on to the next step towards achieving generalizability. So you want to combine the data from the different study sites, and challenges immediately arise. Who has relevant data? How long will it take to get the data? And time and securing the funding is a completely different hurdle. So you see that obtaining the raw data presents its own set of challenges. And now that you have the data, you're faced with the problems of reconciling the different data sets. So data being stored in different ways, data being coded in different ways, data of different quality. So we're dealing with a whole diversity of data ecosystems. And this is where metadata plays a crucial role. So metadata is information about the data, and it provides important relevant context and details. So for example, it specifies the method by which an individual was being aged or sexed, or in this example, it tells us that F4L stands for 4 arm lengths in millimeters. And I can just say that maintaining a comprehensive data set should be common practice, so really do yourself the favor of making sure that your data is well annotated. But sharing your data's metadata makes your data become fair. So it helps making your data become findable, accessible, interoperable, and reusable. So to summarize, the main steps toward integrating data from different study sites are you've got to find data and preserve contextual information in the form of metadata. And you've got to make the data easy to use via standards. And so these standards are ideally already being taken care of in the data collection phase. So data collection already should follow some sort of protocols and data format conventions. And so these are two notable bottom-up initiatives relevant to this particular audience. So there's Euromammals, which has been pioneering collaborative research in spatial animal ecology since 2007. And there's Spybirds, the network that was established in 2019. And both demonstrate how effective these steps are for collaborative research and data sharing. So I don't have the time to explain the process behind Spybirds in detail, but briefly, Spybirds helps with harmonizing data from different studies using code-based data quality checks and standardization. And you can read more about Spybirds in this paper that I linked here. And excitingly, we're currently expanding Spybirds functionality. So we're currently populating a code repository for data processing and data analysis code. And looking ahead, Spybirds is aiming to become a widely recognized, trusted repository for data and code. And so the infrastructure of Spybirds could potentially be used to link a wider variety of data too. So Spybirds is currently mainly set up to host for life history traits, such as laying date or clutch size, as well as some habitat information. But it would be great if the behavioral community could come together and help standardize animal behavior, like other behavioral data, to be integrated with Spybirds. So I hope that the benefits of initiatives like Spybirds are clear, both for those who collect long-term data on individuals, but also for the researchers who want to use it, and for the broader scientific community. So just to summarize, it is easier to find relevant research and to be found. The data and metadata are being archived in a standardized format, and these standards mean that the standards of the protocols mean that the data is of very high quality, and this ultimately saves everybody valuable time. And all of this together facilitates collaboration among researchers. And Spybirds has already an impressive reach. So we're still actively growing the network, but as of June 2022, Spybirds holds metadata of 123 species as studies. And if you decide to join Spybirds and make your studies findable, you can use the QR code on this slide. And for now, all we ask you to provide is the metadata. So basically, which species, how many years, and really, it only takes a couple of minutes to enter this. I want to highlight here that Spybirds is basically it acts as a facilitator. So it provides a service that connects those collecting the data and those wanting to use the data. So yeah, those who collect the data is what we call the data custodians. So basically, Spybirds, to be just very clear, Spybirds does not own the data. The data still belongs to the data custodians, the raw data. But Spybirds acts as a facilitator, helping the data custodians and the potential users to communicate. It first helps the data custodians convert the data into community-defined standards, and then makes it easier for potential collaborators to find each other. And Spybirds is a valuable resource for addressing various questions in animal behavior, ecology, and evolution. So with the data from different long-term studies that have been added to Spybirds, there have been a few comparative studies conducted already. So for example, we can address questions like how the timing of reproduction responds to environmental factors, or how species are impacted by environmental change like climate change or urbanization. And there are a couple of publications that use Spybirds data already linked below. So finally, I invite you to contribute to Spybirds and make use of the resource also. And maybe there's someone in the audience who thinks, I would actually quite like to know what's going on in Shadow Grove Woods. Let's establish Spybirds. So with this, all I have to say is thank you for your attention. Do you have any questions? And please join Spybirds. Thank you.
Thank you very much for the opportunity to present our work here today. A whole range of different fields are concerned with quantifying animal behavior, okay? From applied fields like biomedical sciences, conservation or animal welfare sciences, through to the kind of more blue sky things which I think many of us in this room do, such as behavioral ecology, evolutionary ecology, or comparative cognition, for example. One broad sort of overreaching approach that we all have in common is that we, at the start of our study, define some population of interest, okay, at whatever scale that may be that we're interested in. And typically we draw individuals as test subjects from that population in order to make some wider inferences about the behavior of a larger population, okay? So we study these individuals, and based on our findings, we can extrapolate back to the wider population to make inferences about the biological basis of the behaviors that we are studying. And in an ideal world, the samples that we draw are large enough and diverse enough to capture the full range of behavioral variation which is present within that population. But there are going to be occasions when that doesn't happen, okay? When certain individuals with certain behavioral types are perhaps over or underrepresented within the population. And this could be a problem. And this can come about for a number of different ways, okay? It may arise as a function of the sampling methods that we use to actually capture or collect the animals which take part in our studies, which might target individuals with certain behavioral traits. It might come from how we house the animals prior to testing them, okay? So whether or not we house them in socially or physically enriched environments, for example, before testing them, okay? And it can also be affected by the way that we design our experiments, for example, when we schedule these experiments relative to the animals' circadian cycles or breeding cycles or other aspects of time which affect their behavior. Okay? And if we do this, and if we're not aware of these problems, or if we don't acknowledge them, this is potentially problematic for a number of reasons. Okay? One, because this affects the behavioral data that we obtain, and this frustrates and complicates comparisons between seemingly, on the face of it, similar studies. Okay? And it also limits our scope to generalize from our findings back to the wider population that we're studying or back to other populations. And it also contributes to problems surrounding replication, okay? It hampers replication efforts. So to understand where this behavioral variation comes from, we know that behavior is shaped by the interaction between genes and development and the experience or learning opportunities that the animals had. But in practical terms, when we're thinking about sampling, there's actually a broader range of categories which might contribute to these kinds of sampling biases. And so a couple of years ago, my colleague Christian Retz and I set out in a paper to try and capture the scope of these sampling biases. And we came up with this acronym STRANGE, which we hope captures many of the different things that we're talking about today. So STRANGE covers the social background of the animal, okay, whether it's a social animal, whether it is an animal which lives in a group with a dominance hierarchy, whether it's tested in a social context or not. Trappability and self-selection, so how likely that animal is to end up in our pool of study subjects, okay, or how likely it is to opt into experimental designs where the animals have a free rein to interact with apparatus or not. The rearing history of the animal, whether it's taken from a wild, raised in captivity, enriched or not, and so on. Acclimation and habituation, so this covers how long the animal has to get used to the captive setting or get used to observers who might be watching them in the wild. or get used to the experimental situation once it's placed in an experimental arena, for example. Okay, natural changes in behavior, so changes in the behavior of the animal over the course of a day, between seasons, or over the development of the animal. The genetic makeup, so this applies to differences between populations, but it might also apply to differences between wild type and domestic strains, or between different inbred strains that we might use in our studies as well. And experience, whether the animal has a history of taking part in other experiments or similar experiments previously. Okay, so together these form this acronym STRANGE. These categories are not meant to be super discrete, okay? I'm sure you can all think of different things which fall between these, or maybe things which don't adequately fall within this, but for us the main thing was to come up with this kind of memorable term that would help people think about these problems. Okay, so let's quickly look at some examples of how these kinds of effects might affect behavior. And I want to emphasize the case studies I'm going to go through here. These are actually all examples of well-designed studies where the strange factors were actually the focus of study of the subject, okay? But bear in mind, there are going to be experiments where things like this are also going to affect behavior, but they're maybe not acknowledged, okay? So in this example, the researchers placed apparatus, puzzle boxes, into enclosures containing monkeys, and they had data on the personality traits of these monkeys. And the monkeys were free to interact with the task boxes and generate data as they did so. And they found that the more open and less assertive monkeys were the ones which are more likely to take part in the experiments and generate more data to the data set. Okay. In this study, researchers tested the aggregation behavior of these flies, and they tested them at different phases of the animal's diel cycle. Okay. And they found that those which were tested in the light tended to aggregate more closely. And they also found those which had mated recently also tended to aggregate more closely. Okay, and one final example, these researchers looked at the activity of guppy fish in novel environments, and they compared domestic and feral strains. Okay, and they found that the feral ones tended to be more active, but there's also an effect of acclimation time as well. So the longer you give the animals from both populations to acclimate to the environment, the less active they tend to be. Okay. So these well-designed studies, I'm not picking on these individual studies, there's absolutely nothing wrong with them. Okay, the strange behaviors were the focus of interest, but imagine if these effects were overlooked, okay, if we're not aware of these effects when we're designing our experiments or when we're trying to compare our replication attempt to an original study and we don't get the same results, okay? Just how much variation can factors like this account for in behavioral data sets, okay? And this leads to a following question, and how can we, where we do find differences between studies, how can we distinguish between true biological or ecological or evolutionary differences between populations versus these kind of strange artifacts which arise from the way that we sample the subject pool or design the experiment? Okay, and so in the Strange paper, we encourage people to think about these and to transparently report these different kinds of factors, okay? And critically to tie any conclusions which are drawn to the population of interest, okay? And so we suggest that researchers, ourselves, should routinely ask ourselves, are my test subjects strange with respect to the wider population that I'm trying to understand, okay? Are the test subjects that I'm testing actually representative of a population whose behavior I'm investigating? This is a little bit of how it works in practice. We came up with this 3D design, declare, discuss scheme, okay, to cover this. And so we encourage researchers at the design stage to think about these factors, try new sampling protocols, tweaks to experimental designs which encourage participation, to try and incorporate as much behavioral variation from the population as possible into the study sample, okay? And you'll notice I've put the ARRIVE logo up here, okay? So ARRIVE already covers some of these things, okay? And we've added a few things which we thought were missing, which were particularly relevant to animal behavior. And so this aspect of Strange is designed to dovetail with ARRIVE, okay? One important thing is that we ask researchers to think about these attributes not just with respect to the animals which make it into our final data set, okay, but also those animals which had to be excluded for any reason, which didn't reach training criterion or which failed to engage with the experimental apparatus. This information, importantly, should also be included for those as well. And then in the manuscript, we should all declare an evaluation of our test subjects' strangeness. What do we mean by this? Okay, this is where we can discuss any sampling biases, okay, in our methods which might have affected our results, which were unavoidable, which we couldn't eke out at the design stage, and also include any steps that we took to mitigate these as well, okay? And finally, when we're discussing our work, we should think about just how far we can extrapolate from our findings, okay? We should think about how these different factors might have affected our subjects' behavior, and we should take care not to overreach, okay, and to tie our conclusions as close as possible to the pool of animals that we actually studied. Okay? The Strange is designed to be light touch and non-prescriptive. Nobody likes being told what to do by the likes of people like me, okay? That's totally fine. And we're already overworked as well. Nobody likes checklists, okay? So the onus is on the authors to decide what to declare and what to discuss, okay? And what we're seeing in practice is that in most cases, authors are able to achieve this with a couple of lines in the methods and in the discussion of their papers, okay? And it's also pragmatic, okay? So we recognize that there are reasons why samples often have to be non-representative, okay, for ethical, financial, or logistical reasons. And so we're not setting out to penalize work which is subject to those kind of limitations. All we're advocating for is a kind of transparent reporting of potential limitations. Okay, this is the last slide. Determined to get that chocolate, okay? Now, we've been gratified over the last few years to see that Strange has been taken up by a number of different journals which recommend following the Strange reporting guideline to their authors. And it's also been supported by a number of advocacy organizations as well, covering things like reproducibility and evidence-based animal welfare. And so that's really great. But we're looking to kind of develop this a little bit further as well. Okay, so looking forward, I think there's a lot of scope for meta-analysis here. Okay, there's a wealth of published work out there, and there is scope for meta-analyses which kind of dig into the methodological underpinnings and the sampling underpinnings of this work to try and better understand and quantify the impact of these sampling designs on experimental outcomes. Okay. There's also scope for dedicated experimental work as well to systematically understand how these things work. Okay. And the many studies that we've heard about here already today is a really great way of doing that. They're already doing that. And so I think there's scope to kind of integrate Strange a little bit with that kind of many approach. Okay. We're also looking at producing training resources and reporting and pre-registration guidelines as well. Okay, so in closing, I'd like to say that this is something that we're running at the moment as a side project. It's not something that we're funded for. There's a small core team of us, Kat, Ellis, Christine, and myself. Ellis and Christine are both here as well. And so if you're interested in hearing more about this, working with us, or finding ways to kind of contribute to this, please do come and see us and have a chat. We'll be very happy to discuss this with you. I think I'm very much out of time. Thank you.